Good morning, everyone, to this glorious day. The galaxy of intellectuals, honorable guests, benevolent teachers, beloved students, and everyone present here. How lovely is this day! The scent in the air clearly smells of liberty, kindness, humanity, and everything precious and beautiful. Welcome to the Bacteriophage Enemy and Ally virtual webinar. I'm super excited to have you all here, excited about all the amazing conversations that we are going to have today. Prayer is an action. It's the process of talking to the God with energy in order to achieve a purpose. So let us begin this event by seeking the blessing of the Lord Almighty with a prayer song. I now request our senior PG students to grace this occasion with a prayer song.
जय गणेश जय गणेश जय गणेश पाहि मां जय गणेश जय गणेश जय गणेश रक्ष मां जय गणेश जय गणेश जय गणेश पाहि मां जय गणेश जय गणेश जय गणेश रक्ष मां मुदाकरात् मोदकं सदा विमुक्तिसाधकं कलाधरावतं सकं विलासि लोकरक्षकम् अनायकैकनायकं विनाशितैव दैत्यकं नटाशुभाशुनाशकं नमामि तं विनायकम् नटेतरातिभीकरं नवोदितार्कभास्वरं नमस्सुरारिनिज्जरं नटादिकापदुद्धरम् सुरेश्वरं निदेश्वरं गजेश्वरं गणेश्वरं महेश्वरं तमाश्रये परापरं निरन्तरम् समस्तलोकशङ्करं निरस्तदैत्यकुञ्जरं दरेतरोदरं वरं वरेभवक्त्रमक्षरम् कृपाकरं क्षमाकरं मुदाकरं यशस्करं मनस्करं नमस्कृतं नमस्करामि भास्वरम् जय गणेश जय गणेश जय गणेश पाहि मां जय गणेश जय गणेश जय गणेश रक्ष मां जय गणेश जय गणेश जय गणेश पाहि मां जय गणेश जय गणेश जय गणेश रक्ष मां
Thank you, girls. I now request our professor, Dr. Ramchandra sir, to give the welcome speech.
Good morning to all. The Department of Dairy Microbiology, Dairy Science College, KVFSU, Bangalore is organizing a webinar on Bacteriophage, enemy, and Ali. As we know, the COVID virus, which has shaken the entire world, is an enemy of mankind. Like us, the small living bacteria also gets infection by these viruses. The virus which infects the bacteria is called Bacteriophage. The starters of the bacteria used in the preparation of fermented milk products like dahi, yogurt, cheese. The phages infect these starters and affect their performance also in fermented milk products. Hence, they are enemy to them also. On the other hand, these phages can also be used for the benefits like transfer of genetic material and application in antibiotic-resistant bacteria. So the Department of Dairy Microbiology is also running a project sponsored by DSM, Netherlands, on the detection of bacteriophages in the starter cultures used in fermented milk products in various industries. This is particularly to the DSM starter cultures used in the various food industries. On this background, we are organizing a seminar on the Bacteriophage, enemy, and Ali. So to this webinar, I would like to welcome all the guests. Our Honorable Vice Chancellor, Dr. H.D. Narayanaswamy, supposed to join the webinar due to his busy schedule. He was unable to attend join the seminar. I welcome Dr. Narayanaswamy sir to this webinar. I also welcome our beloved Registrar Sir, Dr. K.C. Veerana, to this webinar. Our Director of Extension, Dr. N.A. Patil, will be joining the seminar in the later stages. I will welcome Dr. N.A. Patil Sir also. And Director of Research, Dr. B. Sivaprakash, will be joining the webinar in the later course. I would like to welcome Dr. Sivaprakash also to the webinar. And we have with us our beloved Dean, Dr. Sachindra Babu Sir. I would like to welcome Dr. Sachindra Babu Sir to the webinar. Thank you, sir. And also we have Dairy Science College Division Head, Arun Kumar Sir. I would like to welcome Dr. Arun Kumar Sir to the webinar. And we have today's webinar is having eminent microbiologist and eminent speakers like our mentor and guide, Dr. P.S. Shankar, is already joined the webinar. I would like to welcome Dr. P.S. Shankar Sir to the webinar. And we have eminent speaker from the NDRI, retired S.K. Thomas Sir. I will be joining as a speaker to this webinar. I welcome S.K. Thomas Sir to the webinar. I also welcome another scientist from the NDRI, Pradeep Behere, to the webinar. Will be joining as a speaker in the webinar. I welcome Pradeep Behere also. And I will welcome Vishnath Angdi from the Agriculture University, Bangalore, Hassan Food Science College to the webinar. So the webinar is organized with the effort of all the department staff. Dr. Prabha Madam has taken key interest in organizing this webinar. I will welcome Prabha Madam and other staff members, Chaya Devi, Lingappa, Nagesh, and all to the webinar. And our PhD and PG students, particularly Rajshekhar and other PG students, have given hands in organizing this webinar. I welcome all the PG students. I also welcome all the college staff who has given help in organizing this seminar. I welcome all the college staff to the webinar. And lastly, I would like to welcome all the students who are joined the webinar online, offline. I welcome all the students to the webinar. And thank you once again, one and all, to this webinar.
Thank you, sir. It was really a warm welcome. On behalf of everybody, I extend a warm welcome to you as well, sir. Light is a symbol of brightness and prosperity, as sunlight expels the darkness of night. Similarly, blessings bring in our life prosperity and happiness. To make this day a blessed one, let us invoke Goddess Saraswati by kindling the lamp of knowledge and wisdom. For seeking the choicest blessings, I would like to invite our university key officers for the virtual lamp lighting. Dr. Narayan Swami sir, Dr. Veerana sir, Dr. N.A. Patil sir, Dr. B.V. Shivaprakash sir. I hereby invite our Dean, Dr. Sachindra Babu sir, and Division Head, Dr. Arun Kumar sir, and the organizing committee for it. Thank you all. We are sorry to inform you that our Honorable Vice Chancellor of KVA FSU, Dr. Narayan Swami sir, is unable to join us today due to his busy schedule, but then he has sent a message for us. I request Dr. Ramchandra sir to share it with the audience.
Our Honorable VC, Dr. H.D. Narayanaswamy sir, was unable to join and address the webinar. He has sent the message. I'll read out the message of Dr. Narayanaswamy sir. The message is like this. Dairy industry is facing problem with respect to starter failures. Lactic acid bacteria are used in the preparation of curd, yogurt, cheese, and these starters may become inactive due to intrinsic and extrinsic factors. And these intrinsic factors may be plastic-borne characters, while one of the major extrinsic factors may be these bacteriophages. They cause threat to starter bacteria if they infect and lyse them, so acid formation is majorly affected. These phages are virulent phages that take up lytic life cycle. The phages are the invisible ultramicroscopic structures and are nothing but viruses. The present webinar is on bacteriophage as danger or beneficial. Both phases need to be known. It is a good attempt from the Department of Dairy Microbiology to arrange such a webinar, which may be talk of the day. I know these phages are now used in therapy called phage therapy, especially against the antibiotic-resistant pathogens to protect from the pathogens effect in human beings. The talks for some of the speakers, as I saw in the program, like Dr. Shankar, renowned dairy microbiologist, Dr. S.K. Thomas sir, and Dr. Pradeep Behere from NDRI are worth to listening. I wish all the success for the webinar.
Thank you, sir. May I please request our Honorable Registrar of KVFSU, Dr. K.C. Veeranna, sir, to address the audience. Guess it will take some time for him to connect with us. We shall proceed with the next guest and get back to him once he is ready. Our Honorable Director of Extension of KVFSU, Dr. N.A. Patil, sir, is unable to join us today due to some technical issue. He has a message for us. I request Dr. Malashri Madam to share it with the audience.
Good morning, everyone. On behalf of Director of Extension, Dr. N.A. Patil, sir, I am here to read out his sent message. And the message says like, the viruses that eat bacteria are the bacteriophages. They require bacteria as a host for their multiplication. And they are dangerous in fermented milk industry where they produce yogurt, curd, shrikhand, and so on, affecting production further to economic losses. Once they are present, cannot be controlled and cannot achieve also. So you can call them as unwanted guests in dairy industry. Further, I have read about bacteriophages as friends killing antibiotic resistant pathogenic bacteria that are difficult to control. As well, they act as scavengers to clean up the sewage if bacteria population is outnumbered. So the Department of Dairy Microbiology, Dairy Science College, KVFSU, Hebbal, Bangalore, initiated the webinar on bacteriophage, an enemy and ally, today on 20th January 2022, with famous speakers having vast knowledge on this topic. I take this opportunity to congratulate the team for conducting such a wonderful, fruitful webinar and wishing them success. Thank you, sir. Thank you.
Thank you, ma'am. The Director of Extension of KVFSU, Shivprakash sir, is on an official travel and is unable to join us today. He has sent a message for us and it goes like this. Bacteriophages are the viruses that kill bacteria. They are considered as uninvited guests in fermented milk industry that produce curd, yogurt, probiotic milk, and many more. The lytic phages destroy lactic bacteria based on their specificity affecting production of FMP. This leads to inability to cope up with supply to customers. They are airborne and require live hosts like bacteria for their survival. At the same time, these phages may act as boon against the antibiotic-resistant bacterial pathogens like Listeria monocytogenes, Staphylococcus aureus by destroying the generation, protecting from causing the disease, and also may act as cleaning agent to control bacterial population in sewage. The Department of Dairy Microbiology Dairy Science College, KVFSU Hebbal, is conducting webinar on Bacteriophage, enemy and ally today. The list of great orators like Dr. Shankar Sir and NDRI scientists having great exposure to such fields seems to benefit the audiences. I congratulate the team for conducting a technical webinar and wish them success. Thank you. Now I request our beloved Dean, Dr. Sachindra Babu Sir, to address the audience.
Very good morning, Honorable Vice Chancellor, Dr. S.T. Narayan Swamy Sir, and Honorable Registrar, Dr. K.C. Veeranna, Director of Research, Dr. B.V. Shivprakash, and Director of Extension, N.A. Patil. The coordinator of the program, Dr. Prabha Madam, Dr. Ramchandra, and head of the division, Dr. Malla Madam, and all the PG students, and my guru, Dr. P.A. Shankar, and the senior faculty, Tomar Sir. And first of all, I place on record the Department of Dairy Microbiology, which is doing a wonderful work. And in this pandemic time, this is the second time they are organizing a webinar. And the webinar on Bacteriophage's enemy or friend is nearly need of the hour. And I congratulate them and also place on the record this thing. And once the BPs, popularly known as BPs, I'm not an authority to talk about BPs, but even though I'll tell you a few things, because all in front of authorities, I can't speak about the microbiology. BPs once considered to be a nuisance in the dairy industry and responsible for fermentation failure and economic losses. There were a lot of economic losses, and in fact, now it has become very promising, and it has become to kill all the microbial pathogens. Okay, and this is a day we have to also remember the Nobel Prize winners like Max Delbruck, Alfred S.C., Salvator, who have done a lot of work to this BPs. And BPs are not only in the dairy sector, it is also becoming an alternative medicine to human beings. And once upon a time, it is said that it is a nuisance. Now, it is here to cure all the chronic ear diseases, infections, burns, wounds, cystic fibrosis associated with lung infection, pneumonia, and cystic fibrosis, and positive blood culture also can be done. And now here I can remember the Tom's story. I don't know whether any of you have watched that Tom story. His wife says that my husband has been saved from sewage, from superbug. And almost he was chronic and he was bedridden with severe pancreatic, and he was in total deathbed, and all his related organs were failing. I'm just supporting that it is the benefits of the thing. Or almost he was in a deathbed, and she was the one. She was not a medical doctor or nothing. She herself on her own, she suggested the medical professions that it can be cured by pages. And finally, within 1 or 2 days, he got up. And she was so happy, and it is in YouTube also. You can watch it. It's on Tom's story. And also like that, there are a lot of case studies which are happening, and also, but this has come to our rescue to have a safe and food products also. And for your information, LMP 102, ready to eat, USA has released. These are certain things which, and also not only this, it also will help in our counteracting our bioweapons, toxins such as anthrax and botulism. Botulism is another threat, Botulism can be handled with this type of thing. Not only this, even it can be used for horticulture, protecting our plant, and other things. Apart from that, even hospital industry are also into pages, especially the catheters. Catheters, you may be knowing that is cardiac catheters are there, urinary catheters are there. All such things have been used by all the medical devices, sutures, and almost all surgery. And also, let me not take much of time. There are a lot of stories on pages wherein even a heart where transplant was done for a 80-year-old person. Like that, and there are many success stories. And coming to livestock sector, since we are from livestock sector, it is also very much used in treating all the pet animals and dairy animals and other animals. And it is also not only that, it is also becoming one of the measuring yard for a healthy person. It is said that around 62 to 54% of the thing, if pages are there, it means in a gut, mean to say that he is a healthy person. As it comes down, it is understood that he is a unhealthy person or he is becoming older. And it is not only from all of a sudden we don't get it. We get it through our mother itself. From the mother's womb itself, as soon as we take birth, we get these pages. And it is not only found in human beings, even in primates also it is found. So this is information which I want to share with you. With this, we'll listen to our Thomas sir and Dr. P. A. Shankar sir, so who are the authorities, and Prabha madam and Dr. Ramchandra. With all this, I congratulate the Department of Dairy Microbiology, Dr. Prabha madam, Dr. Ramchandra, Mala madam, and Chaitra, especially. Thank you, Chaitra, for nice Rashekar and all the PG students. Once again, congratulate all of them. Thank you very much. Thank you.
Thank you, sir. I call upon Dr. Malashri Madam to propose the vote of thanks.
It's my privilege to propose vote of thanks. I would like to express my gratitude to all the esteemed delegates of today's webinar, Vice Chancellor of KVFSU, Dr. H.D. Narayanswamy sir, Registrar, Dr. K.C. Veeranna sir, Director of Extension, Dr. N.A. Patil sir, and Director of Research, Dr. Shiv Prakash sir, for taking part in this inaugural function, and also thankful to their kind wishes to make this webinar a great success. Gratitude is not in the words, but in the heart which expresses it. So with this token of gratitude, the entire Dairy Microbiology Department and DSM team showers sincere gratitude to our beloved Dean, Dr. Sachindra Babu sir, for his presence in inauguration of this webinar. Also, I thank our beloved Division Head, Dr. Arun Kumar sir, for his kind support. I also thank all the eminent speakers of today's webinar, participants and students for taking part in this webinar inauguration, and also I wish good success to this webinar. Thank you.
Thank you, ma'am. Let us now move to the technical session. Thank you, sir.
Thank you, sir.
Thank you. Hello everyone. Please give us some time as we connect with all the speakers for the technical session. Welcome back. Our registrar, Dr. K.C. Veerana sir, is unable to join us today, but then he sent a message for us. I request Dr. Ramchandra sir to share it with us.
Our Honorable Registrar Sir, Dr. K.C. Veerana sir was unable to attend join the webinar. I just read his message to the webinar. Since I launched the joining of the webinar, I wish all the best to the team of Department of Dairy Microbiology. They are conducting the webinar on the bacteriophages, which they infect and kill bacteria, and their presence affect lactic acid bacteria in getting the fermented milk products. So with this background, I congratulate the team of Department of Dairy Microbiology for conducting the useful webinar on the bacteriophage, enemy and Ali. Thank you.
Thank you, sir. Let us now move to the technical session. Bacteriophage as an ally or bacteriophage as an enemy. Just as death in life, sorrow and joy are the two sides of the same coin. And therefore today, we shall review these two sides of the bacteriophage. So today our speakers will give you an insight about today's topic. Without any further delay, let's get started. I request Dr. Ramchandra sir to brief about our inaugural speaker, Dr. Shankar sir, to the audience.
Dr. P.A. Shankar sir, retired as Professor and Head of Department of Dairy Microbiology in the year 2006. He holds Master's degree in Dairy Microbiology from the Great Institute, National Dairy Research Institute, Kernal, and he obtained doctoral degree in Dairy Fermentation Technology from Reading University, England. He served as first Director of Instruction, Dairy Science of Dairy Science College, University of Agricultural Sciences, and he was the first Director of Instruction postgraduate studies when the newly Farm University, Karnataka Veterinary, Animal and Fisheries Sciences University at Bidar. He was getting the first Director of Instruction, Postgraduate Studies. He is a recipient of ICR Junior Fellowship for Masters and he had Commonwealth Fellowship for the doctoral degree. He was Principal Investigator of ICR funded and DBT funded research projects. He has conducted various research projects which are useful for the department as well as for the industries, and he guided several masters and doctoral scholars. He published several research papers at the national and international level, and he also published various technical articles, and he attended several scientific conferences at the national and international level, and he was one of the key person, and he was involved in the expert as an expert for the National Dairy Research Institute, Agriculture Science Research Board, UPSC, and he was the member and committee member of the several universities. After his retirement, he served as director, R&D facility on activated carbon-based water purifiers with focus on nanometals, including nano silver. Presently, freelance advisor cum consultant. Now, sir is staying in Australia with his children, and I hope he is keeping his well very well. And now I request Dr. P.S. Shankar sir to deliver a technical session, first technical session in this webinar. Sir, you will run slides yourself or shall we run, sir? Sir, mute आइए. Shankar sir, unmute ಮಾಡಿ. Sir, mute ಆಗಿದೆ sir. Unmute ಮಾಡಿ sir.
Yes. Okay. Can you hear me now?
Yeah, yes sir. Sir, you'll run your slides or shall we run, sir?
No, I'll run from my side.
Okay, sir. Thank you, sir.
I'll run from my side.
Welcome, sir.
Thank you.
Yes, sir, we are able to see.
Okay. Can you see?
Yes, sir. Yes, sir. Clear. Yes, sir.
Okay.
Thank you, sir.
Thank you very much, Dr. Ramchandra, especially for introducing me to the new audience, especially students and postgraduate scholars. And you are right, I am in Australia, and we are 5.5 hours ahead of India, which means we are in the afternoon session, post-lunch session. But still, I'm more than happy to participate in the webinar. At the outset, let me congratulate Dr. Prabha and her team for organizing this webinar. On bacteriophages, enemy and ally. And it's indeed an important topic to be discussed and talked about for many reasons. One of the reasons would be to motivate youngsters, especially postgraduate scholars, to think of doing something away from what has been done so far in the department, of course, elsewhere, so that we'll have a wealth of knowledge on bacteriophages. And I also would like to thank Prabha and her team for inviting me to take part in the webinar, although I express my reservation as I'm out of touch with many of these topics. But still, I thought, let me share whatever few thoughts I have on this particular topic. So I've chosen the title of my presentation as an overview on bacteriophages with focus on bacterial immunity to phages, which is something which I found important as all fermentation microbiologists always pray that their cultures don't become victims of phage attack. Every day when they come back to the laboratory or to the production unit, first thing they do is to see whether the culture has grown or not. That means whole night they might be wondering whether my culture grows or not, whether it is attacked by a phage, or what happens to my culture. So I thought, let me share my thoughts on how to make this bacteria immune to phages so that bacterial fermentation or microbial fermentation can go on without any hindrances.
Sir. Good morning, sir.
Okay, I'm coming. Next slide.
Good morning, sir.
Good morning.
Sir, Rudraya from Monday Milk Union, sir.
Okay, thank you.
Second.
Thank you for joining.
Second batch, sir.
Okay,
PT.
thank you for joining.
Thank you, sir. Thank you.
First presentation, introduction. Go back. I'm glad to note that some of our alumni also joined, and joining probably. And let me introduce my presentation. The focus, as I mentioned, is on how to develop resistance or immunity in bacteria against phages. For those of us who need to refresh their knowledge or information on bacteriophages, let me recap some basic points so that especially undergraduate students may be benefited. Phages are obligate intracellular parasites. That means they cannot grow outside the body, they cannot grow on their own. They need bacteria in order to multiply or survive. So therefore, they're very much dependent on bacteria to survive. But interestingly, bacteriophages are much smaller than bacteria they destroy. Normally, one would think the one who is destroyer is more powerful than the victim. But in this case, it is the opposite. The phages are smaller, whereas bacteria are bigger, but phages can still kill bacteria. Now, interestingly, phages are distributed in the entire biosphere. It is not that a particular area is free from phages, because wherever bacteria exist, phages also exist. So in the evolution process, they become co-evolution. Both bacteria and phages have evolved together. So wherever large quantities or reservoirs of bacteria exist, such as soil, as animal scientists, we know rumen is a big source of microbes, whereas non-ruminants have large intestines as a big source of microbes. So you find in all these natural reservoirs both bacteria as well as phages. But in the nature, it appears seawater is the densest source of phages, because in sea water varieties of microbes exist, and therefore naturally a variety of phages also exist there only. So of late, of course, there has been some attempts to use them as alternative to antibiotics. I'll come back to that later. If you look at how the bacteriophages look like, shape or appearance, they're composed of nucleic acid material surrounded by a protein structure. Now, these phages must first attach themselves to bacteria. Without attachment, they will not be able to kill the bacteria or multiply their own progeny. So therefore, if there is a bacterial infection, it must be attached by a phage. So even here, the selection of phage and bacteria are not random. It is always very specific and unique. Only those bacteria which have unique character on the cell wall, which we call phage receptor, will be able to become a victim of phages. If that receptor does not exist on bacteria, phages will not be able to attack. In fact, this is one of the mechanisms bacteria employ to resist themselves from the phage attack. Now, when the phages attack bacteria, it is not the entire phage enters into the bacteria. No, it's only the nucleic acid of the phages enter the bacteria. So therefore, there must be a mechanism in the phages so as to push the phage DNA into bacteria. How it happens and all those things, we will discuss about that. And when it does push the nucleic acid into the bacterial cell, sometimes it is called as transduction. We will discuss more about transduction as we move forward. Just to recap again, having said so much about phages being small, tiny, one would like to know what is their size, how big, how small, how they look like. So we'll start looking at the phage size. It's approximately 200 nanometers long in the case of T4 phage, which is considered to be the largest phage. But otherwise, most of the phages have size ranging from 24 to 200 nanometers in length. Can you imagine how small they are? Now, these phages have a head or capsid, and the head structure can vary in size and shape. Some are icosahedral and others are filamentous. The head is composed of many copies of one or more different proteins. Inside the head is found nucleic acid. When it comes to tail, many but not all phages have tails attached to the phage head. The tail is a hollow tube through which nucleic acid is pushed through. In the more complex phages, the tail is surrounded by contractile sheath and spike and so on and so forth. So also, the last structural component of the phage is a base plate. At the end of the tile is a base plate. One or more tile fibers are attached to it. Both the tile plate and tile fibers are attached to the binding site. As I said earlier, unless it attaches to the bacteria, it will not be able to infect the bacteria. So therefore, phages have a mechanism to attach themselves on bacteria. What all I said now is now pictorially presented, such as capsid head, which contains nucleic acid. And I also said a tail, which has a collar and sheath. I also mentioned about base plate. Then, of course, spikes and tile fibers. This is considered to be a very classical shape and structure of bacteriophage. If one looks at any publication on phage, one finds this beautiful picture giving all the components of the phage. It does not mean all phage particles contain all of them. We shall see how they look like. It is not an imaginary structure. It has been photographed using electron micrograph to see how it looks like. So therefore, one can see physically through electron micrographs the appearance and shape of phage. Now, as I said in the beginning, or rather earlier, phages have different shapes. Some of the shapes I mentioned here, including the classical one, but also other which almost look like a spindle, a blob, a square, all sorts of shapes one can imagine. As I mentioned earlier, bacteria also are not of the same uniform size. All bacteria don't look like E. coli, nor staphylococci, nor streptococci. Bacteria have different shapes. So also to match with these shapes, phages also have different shapes. As I said earlier, it's a co-evolution. That means as bacteria grew to different shapes, phages also grew to different shapes to match with the shape and size. Now, once the phage enters, or phage DNA enters the bacterium, as I said, multiplication of the phage begins to take place. Remember, only nucleic acid has gone in, not the entire phage has gone in, which means whatever nucleic acid that has gone in, either it can be DNA or RNA, it must go through different stages so as to form phages inside the bacteria. How it happens and what are the stages involved, we'll go through it. For convenience sake, we group them into multiplication through a process called lytic cycle, or lysogenic cycle. In the lytic cycle, sometimes referred to as virulent infection, the phage kills the host cell to produce many of their own progeny. After the nucleic acid is injected into the host, the phage synthesizes proteins that break down the host DNA. Remember, the entire phage that has only DNA has gone inside, entire phage has not gone in, which means it must synthesize the whole phage. Remember, you saw the picture. So, using single nucleic acid, the whole structure has to be synthesized within the bacterium. And therefore, it uses the bacterial cellular machinery to synthesize each and every component of the morphology. For example, heads and sheaths are assembled separately. Genetic material is then packed into the head, and new daughter phage particles are constructed. So therefore, as one can imagine, this process is very complex. Several stages are involved, and each of the stages are critical for infection. Finally, when the daughter phages are made, bacterium is burst open, and nearly 100 to 200 new phage progenies are released from each bacterium. I mentioned about lytic cycle. Let us go through what are those steps involved in the lytic cycle. Broadly, they are considered as 4 steps in the lytic cycle. First one is phage attachment, as I mentioned earlier. Unless phage is attached to the bacterium, no phage can multiply. After the attachment is done, bacterial nucleic acid enters the bacteria. Then phage replication takes place. And finally, the birth of new phages takes place. This is shown again in a classical way. First, now one can understand, easily appreciate what all I mentioned earlier. Attachment, bacterium is shown in a very big size. Phage is shown in the classical structure. And you can see how phage is attached to the bacterium, and then how nucleic acid enters the bacterium, and how nucleic acid from phage takes control of the entire bacterial metabolism, cellular machinery, and viral genome is constructed. The host genome is virtually degraded. There is no more bacterial DNA exists. Then viral genome begin to multiply. You can see the replication of that, because viral particle requires both DNA as well as protein. All of them have to be synthesized. Then they must form a shape. That means attachment should take place. And then finally, it should burst open and release the phage particle. This is how it is depicted. Each of the stages are shown in this diagram. Just for some of you who may be wondering all this mechanism of attachment and all, I just picked up one of that and magnified it. You can see the bacterium here. You can see the phage here. Nucleic acid is seen in the head or capsid, both what we call DNA or RNA, and see how it is attachment taking place. Then how the nucleic acid is pushed through the cell wall into the cell. So this is a pictorial way of showing the mechanism in more detailed manner. Now, always people look for some physical evidence. Electron micrographs, you can see on the left side a bacterium where you can see a couple of phage particles attaching. Then you can see in the next one how the bacterium is broken down, burst into, and then phage particles are coming out. So it is not only an imaginary picture, it is demonstrated using some of the electron micrographs. So this is about lytic cycle. Just to recap, one phage particle can enter into the one bacterium, release up to 100 to 200 phage particles. You can visualize what is the number of phage particles that can come out from each and every bacterium. If there are 1 million bacteria, you can imagine it becomes easily 100 million phages. Now 1 million phage is not a big number at all, but you can see the number of phage particles that can come out. 100 million phage particles can come out. Now, the other method is lysogenic cycle, which is also a very interesting mechanism. And it is not that moment phage DNA enters into the bacterium, it kills the bacterium and progeny is generated immediately. Again, in nature's way of protecting itself. a mechanism called lysogenic cycle also exists. And this cycle sometimes referred to as temperate or non-virulent infection. Infection takes place, but it is non-virulent. That means it doesn't kill immediately, does not kill the host cell immediately, but it takes a refugee. That means it takes protection and remains in the bacterium as a dormant state. Now, at some stage, once it is inside the bacterium, it so nicely integrates itself with the bacterial nucleic acid. Then remains as part of bacterial DNA. The advantage being, once it becomes part of bacterial DNA, as bacteria multiply, phage DNA or phage nucleic acid also multiply. That means replication is coexist with the bacterial replication. Because phage genome is considered to be a very small one compared to bacterial genome, but still, because it becomes part of bacterial genome, it remains there, and then but during this process, it doesn't harm the bacterium. It remains as what we call temperate phage, or just takes refuge in the bacterium. However, important component. Now, the same thing is said here in a pictorial manner. Phage attaches to the cell surface of bacterium. Then, as you can see, phage DNA enters to the bacterial cell, then becomes, a new word is coined, is called prophage. It is phage, but it is pro, waiting for some opportunity. Then integrates itself. It doesn't remain separately from the bacterial DNA. It remains part of bacterial DNA. And as bacteria multiply, phage DNA also multiply, which means when bacterial progeny multiplies, one cell becoming 2 to 4, 4 to 8, like that, phage DNA also multiplies, which means it's something like saying, wherever you go, I will also come. If you become monster, I will also become monster. So I am with you all the time. So this is how it adopts that concept of, it's not only one bacterium, but all its daughters, all its phage multiplications. Also we find phage DNA. Again, the same thing is projected in a pictorial way, how a lytic pathway takes place, how a lysogenic pathway takes place. Now, when a phage becomes lytic, when it will become prophage or lysogenic, various factors influence. Now, we will not go into those details now, but just to give you some idea how a lytic becomes lytic, or when a phage becomes lytic. Now, when the bacterium carries a prophage, now you understood what prophage means. Prophage means bacterial DNA becoming part of bacterial DNA without killing the bacterium. It is what we call integral part of bacteria. Now, when such bacterium carrying the prophage or phage DNA is exposed to certain stress factors, such as UV light, low nutrient conditions, chemicals such as metamycin, these prophages spontaneously extract themselves out of the host genome and enter the lytic cycle, and this process is also called induction. A recent work has shown that this transition between lytic and lysogenic infection is also dependent on the abundance of phage in the area, as they're able to produce and sense small peptides in a process akin to quorum sensing. Again, a very complex mechanism. At this stage, we'll not go into all those details, but just to keep you updated that there are certain conditions, and those conditions favor multiplication mechanism. Now, using all these issues, particularly this transduction part, phage is used as a genetic material. Remember, it can become part of bacterial DNA. So one must be thinking, when it comes out of the bacterial DNA, does it pick up any of the bacterial DNA along with it? It's something like if you are eating food along with your friend, and sometimes you put your finger on your friend's food and also eat up. Like that, here, the phage DNA, when it is coming out of the bacterial DNA, it's likely that it may pick up some portion of bacterial DNA also. So this has been proven using various processes, and as early as 1950, in the Salmonella process, bacterium, this mechanism was established. That's how this transduction was considered, which means nucleic acid has been transferred from one to another one. Now, in fact, human insulin was cheaply produced using this phage DNA. Again, we will not go into all those details, which is, again, as I mentioned, those who are in the molecular biology field will be able to understand the role of importance of nucleic acid transfer from bacterium to bacterium, from bacterium to unicellular organisms, and then from unicellular organisms to multicellular organisms. These are a little complex. We'll not go into the details about it. My effort here is to highlight that phages are the starting point for all these complex processes. Now, as I mentioned, that transfer of genetic material is one of the important component of the whole exercise of phage infection. Now, some of this is accidental. Some of this is, of course, obviously with the intention of making phages more robust. So the process is wherein part of DNA is transferred from one bacterium to another bacterium via phage. Sounds interesting, isn't it? That means a phage first infects the bacterium. The phage kills the bacterium, and in the process picks up one bacterial DNA, then attacks another bacterium. And when it attacks another bacterium, it carries the DNA from the previous host and transfer to the next host. Again, little interesting, but not complex anymore. It used to be complex when we were students, but now it's no longer a complex. Now you can see most of them and very nicely demonstrated. So therefore, we can see that DNA gets incorporated into the phage, then the DNA is transferred to recipient bacteria at the time of infection. Now, bacterial donor DNA can be incorporated into the phage by either lytic or lysogenic cycle. Remember, when it is lysis, it carries immediately. When it is lysogeny, it takes after some time. The three types of transduction I explained. One is called generalized transduction, specialized transduction, and lateral transduction. Again, those who are keen to understand all of them can find a lot of publications on this. But for the so-called beginners, just a quick review of what we discussed, that phage attack, and then picks up bacteria, phage DNA, multiplies, breaks up, and then you can see some of the phage carrying the bacterial DNA. This is called generalized transduction. Now, specialized transduction, you can see the color of bacterial DNA different from phage DNA. Can you see? Circular DNA is bacterial DNA. A piece of DNA that is coming from phage is shown as red color. As you can see, it becomes part of the bacterial chromosome, bacterial DNA, becomes integral part. And then, of course, when it multiplies, it also multiplies. And when bacteria are exposed to stressing factors, stressors, then it breaks up, and you can find in those phage particle part of DNA. You can see that some of the phages have a blue nucleic acid, which was not present in the beginning when it infected the bacterium, indicating that this so-called new phage is carrying the bacterial DNA from the previous host. Of late, very recently, people have been examining lateral transduction. All three have been presented here: generalized, specialized, and lateral. Again, I will not go into the details of all these mechanisms, but to convey that there are various systems, mechanisms available through which bacterial DNA and phage DNA merge with the phage DNA, and then phage DNA can in turn transfer to another bacterium. So these are all the mechanisms by which nucleic acid from the phage is transferred. Now, having understood so much about transfer of genetic material from phage to bacteria to bacteria, well, let us look at the other aspect of phage and bacteria. Remember, we said bacteria can kill. So it must be in the mind of some of our curious scholars how to prevent phage infection, how to make bacteria more robust, how to make bacteria immune to phage attack. So these thoughts would be natural. And as I said in the beginning itself, both phage and bacteria are co-evolution. Whenever there is a co-evolution, there is always a fight and mechanism to overcome the fight. In both the cases, there is virtually what we call a race, what we call here arms race. That means you have one sword, I have two swords, you have one pistol, I have another two pistols, you have something else, so that you have protective mechanism, I have a laser machine, I have a laser protective mechanism. So all those competitive mechanisms to protect oneself against the attack is developed, or as I mentioned to you, thousands and thousands of years have caused all these evolutionary changes. Just to bring it a little more structurally, throughout evolution, bacteria have been preyed upon by parasitic bacteriophages. Everywhere bacteria are found, therefore phages also exist. And therefore, there's always a continuous cycles of infection. However, in order to survive and thrive, bacteria have developed a set of arms arsenal against phages through antiphage mechanisms. And the competitive bacteriophage coevolution, often referred to as an evolutionary arms race, has produced a multitude of bacterial defense mechanisms, and these are all inhibit at every stage of the phage cycle. It's not that only it just says, I will not allow you inside. No, it will ensure even if by mistake the phage DNA enters, how does it inactivates, neutralizes, kills the phage DNA. So I'll just help you to mention, preventing phage entry is one mechanism, preventing phage adsorption is one mechanism, preventing DNA injection itself, it can stop. For those of you who are familiar with antibiotic resistance, Remember, you must have read there are some antibiotics which will prevent the multiplication of bacteria. There are some antibiotics which enters the bacterium and then inactivates nucleic acid synthesis. There are some antibiotics which enter the bacterium and prevent protein synthesis. Likewise, similar mechanisms exist here also. Only difference here is where antibiotic doesn't have an immune system, whereas bacteria here, both phages and bacteria both have fighting capabilities. Each of them exhibit their own fighting mechanisms. Then targeting continuing the discussion, targeting bacteriophage nucleic acids, restriction modification system, CRISPR-Cas systems, abortive infection toxin antitoxin mechanism, bacteriophage assembly. That means even after all the nucleic acids are components are produced, the formation of bacteriophage can be prevented. So apart from these known mechanisms, there are so many unknown mechanisms, and each of them just again pictorially presented. So receptor binding can be prevented, silencing the lysogen repressor, then cleavage of nucleic acid, the CRISPR, and prophage is inactivated, then toxin, all that what I mentioned earlier have been pictorially presented here to appreciate. It is not that whatever phage DNA that enters inside can automatically result into the production of daughter phages. There are mechanisms. The reason I went into so detail is to help any of our microbiologists who are working on starter, who want to develop starter resistance against phage attack. They can consider any one of these mechanisms as a possible way of preventing the phage attack. Now, the last one or two slides I'll discuss on phage therapy. Both Dr. Ramchandra and others also mentioned about the use of phages as drugs, like sulfonamides, like antibiotics, which were very, very, very extensively used in the last 30, 40, 50 years. However, of late you must have heard about superbugs, which means they are not easily killed by or inactivated by any of the sulfonamides or antibiotics. What we now call, some of you may be familiar, antimicrobial resistance. So these superbugs are really a worrying factor. So in the last 30, 40, 50 years, where we have used extensively large number of antibiotics, automatically we must have produced superbugs as they are resistant to most of the antibiotics. One can imagine what would happen to an individual. He simply gets an infection, but unfortunately, all the antibiotics are incapable of killing the bacterium. His answer so far has been to pray God that these bacteria die on their own. But people have been exploring the possibility of use of phages to kill or eliminate such pathogens. But this is not something new, which was in practice in early 90s, but both sulfonamides and antibiotics have overtaken the role of phages, and therefore people have forgotten about phages. But of late, because of the bacterial resistance, because of the challenges, new antibiotics are not being developed, because no new antibiotic has been developed in the last 20 years. What all the antibiotics that we are using is almost 20-year-old, because no new antibiotics are developed because of the difficulty in developing the antibiotics. So therefore, phage therapy is becoming one of the latest strategy to fight against the infection. But there are some methods by which one can train, what we call phage training. Again, these are all few challenges. These are all developmental stages. Again, I will not go into the details, except to say that phage therapy, which had been in usage 40, 50 years ago, has now come back, and people are showing more interest in that. But again, it needs to investigate further to study all these mechanisms, understanding specificity. All those issues are there in the phage therapy. So with this, I would like to conclude that we must understand that phages and bacteria are living together for millions of years. There's always been a back and forth arms race between phage and bacteria. And therefore, this must not escape our attention to talk about phages. And phages are extensively used in molecular biology. And there are various strategies developed to prevent the phage attack. But with all that, fermentation industry is always worried about phage attack. And therefore, they always have to monitor the appearance of a new phage. Now, in my conclusion, I had said about phage therapy. While I was going through a newspaper today, I found that as per today's Lancet publication, 1.2 million people are died because of superfog infection. To cure those infections. And 3.68 million people died because superbugs played a very destructive role in their infection. With this, I conclude, and I would like to thank one and all, and a special thanks to organizers for giving me an opportunity to share my thoughts, which I thought I have forgotten are all old. But for PG scholars, I hope this presentation would serve as a sparkle to think outside the box for their research work. I hope they'll take this as forward. Thank you one and all again.
Thank you, Dr. Shankar, sir. Many thanks to Dr. P. S. Shankar, sir. Even though he is in Australia, he has taken a lot of risk to prepare slides, even 20 slides, 20 minutes talk. Thank you, sir. Sir, many of our KMF seniors like General Manager Mun Reddy, General Manager Rudraya, and all your students are waiting to interact with you, sir. And I request all, if you have 5 minutes, if you want to interact with Shankar, sir, you can interact.
I'm more than happy to interact. If there are any questions or...
Yes, sir. Yes, sir.
Yeah.
Sir, I'm Dr. Shankar Lingegoda, sir. Hegidiri, sir.
Thank
you, sir. Now, Chennai, sir. Thank you very much, sir. Sir, I have a small question. Can we predict the bacteriophage attack on culture product? How to predict it?
You don't have to predict because whatever phages you have, you expose it to bacteria. If bacteria dies, that means it's done. That is one method. Another complex method is to expose phages to ultraviolet and see whether there any lysogeny exists and then come out of it. So therefore, one word answer for that is difficult, but it is possible. I think Ramchandra and his group are working on that. They have a project on phages, focus on starter cultures. So they would surely come out with something to screen their cultures for phage presence. How do we screen them? For example, if you give me a culture and ask me to screen whether it is likely to be affected by phage, one of the mechanisms we normally do is to expose to so-called known phages. Known. You give me a culture. Remember, when you go to a doctor and if you have an infection, they take a swab and test it for known antibiotics, which means whether any of the known antibiotics can inactivate that particular infection. Accordingly, doctor will prescribe. Similarly, if you give a culture to Dr. Ramchandra, maybe next year or 2 years later, he will screen, he will expose that culture to various known phages and see whether any phage can attack. If no phage attacks, which means reasonably it is safe. Remember, it is reasonable because I mentioned earlier, if the phage is unfortunately if the bacterium culture is carrying a lysogeny or prophage, then he will not be able to find out. But reasonably, one can say it is not prone for phage attack. I don't know whether you're able to answer this, but I'm sure.
Sir, Sankarlinga Koda sir, we will be discussing detection method in my talk, sir.
Okay,
Very good.
okay. Thank you,
Very
sir.
good, very
Thank
good.
you very much, sir.
Very
Actually,
good.
we are doing DSM Netherlands projects for the detection of phages in the fermented milk products itself for the
Very
starter cultures, but only for DSM culture now we are doing because they are not given to all the other commercial cultures. Only for
No, you
DSM.
can always, you don't have to tell them. Of course, I'm not supposed to talk like that, but still you don't have to tell them. You can ask your PG students to screen
Yes, sir.
known cultures.
Okay, sir.
Non-DSM cultures rather, non-DSM.
We are doing for curd and yogurt.
That's
Like, where samples will be taken. I will be
That's
discussing
it.
detailed detection method in my presentation, sir.
Okay,
Yeah,
okay. Thank you, sir.
DSM.
Thank you very much. You have taken us back to our college days also. Anyway, thank you very much.
Thank you, Dr. Shankar sir. I have become a student again.
Okay, thank you.
It's quite interesting, sir.
Yes,
This is
yes.
Rajaratnam. Thank you,
Yes,
sir.
yes. I know you are there.
Thank you. Thank you. very much.
Thank you. Thank you, sir.
Thank you.
Thank you.
Thanks
Thank
for
you,
your time.
Ramchandra.
Rudraya sir wanted to speak to sir. I think you can speak, sir. Rudraya sir.
ಹೇಳಿ ರುದ್ರ ಚೆನ್ನಾಗಿದ್ದೀರಾ ನಿಮ್ಮ ವಾಯ್ಸ್
Rudraya sir,
ಕೇಳ್ತೀರಾ
unmute ಮಾಡಿ sir. ಮ್ಯೂಟ್ ಆಗಿದೆ, ಅನ್ಮ್ಯೂಟ್ ಮಾಡಿ ಸರ್ ಮೈಕ್ ನಿಮ್ದು.
ಮೈಕ್ ಅನ್ಮ್ಯೂಟ್ ಮಾಡ್ಬೇಕು. ಮೈಕ್ ಮುಂದೆ ಒಂದು ಕ್ರಾಸ್ ತೆಗೀರಿ.
ಓಕೆ ಸರ್. ಸರ್ ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಎವ್ರಿಬಡಿ ಸರ್.
ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಹೇಳಿ.
ಸರ್ ಬಹಳ ವರ್ಷಗಳ ನಂತರ ನಿಮ್ಮನ್ನೆಲ್ಲ ನೋಡ್ತಾ ಇದೀವಿ, ಖುಷಿ ಆಗ್ತಿದೆ ಸರ್. ನಾವೇ ಸರ್ ತಮಗೆ ಗೊತ್ತಿದ್ದಾಗೆ ನಮ್ಮ ಇಂಡಸ್ಟ್ರಿಯಲ್ಲಿ ಕೆಲವು ಟೈಮ್ ಅಲ್ಲಿ ಸರ್ ಈ ಫ್ಯಾಸ್ ಅಟ್ಯಾಕ್ ಆಗ್ಬಿಟ್ಟು ಸೆಟ್ಟಿಂಗ್ ಎಲ್ಲ ಆಗದೆ ಅಥವಾ ಪ್ರಾಬ್ಲಮ್ ಆಗ್ತಾ ಇದೆ ಇಂಡಸ್ಟ್ರಿಗಳಲ್ಲಿ. ಅದಕ್ಕೆ ನಾವ್ ಏನೇನ್ ಕೇರ್ ತಗೋಬಹುದು ನಮ್ ಕಲ್ಚರ್ ಬಗ್ಗೆ. ಈಗ ಆಲ್ಮೋಸ್ಟ್ ಸರ್ ನಾವು ಹೊರಗಡೆಯಿಂದ ಖರೀದಿ ಮಾಡ್ತಾ ಇದ್ವಿ ಕಲ್ಚರ್ಸ್ ಎಲ್ಲ, ಸೆಟ್ ಕಲ್ಚರ್ಸ್ ಎಲ್ಲ. ಸೊ ಅಂತ ಬಗ್ಗೆ ಏನ್ ಕೇರ್ ತಗೋಬಹುದು ಸರ್? ಕೆಲವು ಟೈಮ್ ಏನಾಗ್ತದೆ, ಈವನ್ ಸಪ್ಲೈಯರ್ಸ್ ಕೆನಾಟ್ ಡೂ ಎನಿಥಿಂಗ್ ಅಟ್ ದಟ್ ಶಾರ್ಟ್ ಪೀರಿಯಡ್ ಸರ್.
ಫಸ್ಟ್ ಏನ್ ಮಾಡ್ಬೇಕು ನಾವು ಅಂದ್ರೆ,
ಹಾ ಸರ್,
ಯಾವುದೇ ಒಂದು ಸ್ಟಾರ್ಟರ್ ಕಲ್ಚರ್ ಫೇಲ್ಯೂರ್ ಆಗಿದೆ ಅಥವಾ ಆಗ್ತಾ ಇದೆ ಅಥವಾ ಸ್ಲೋ ಡೌನ್ ಆಗಿದೆ ಅಂತ ಗಮನಕ್ಕೆ ಬಂದ್ರೆ, ಫಸ್ಟ್ ವಿ ಮಸ್ಟ್ ಲುಕ್ ಅಟ್ ವೆದರ್ ದೇರ್ ಆರ್ ಎನಿ inhibitory components in the milk. I think Ramchandra group will help you. ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ, if there are antibiotic residues in the milk, especially in a pooled milk samples, so first thing we should check for presence of antibiotic residues. Very simple, they're all ಈಗ ಸುಲಭ ಮಾಡ್ಬೋದು ಇವನ್ನೆಲ್ಲಾನು. ಸೊ ಇವನ್ನೆಲ್ಲ ಮಾಡ್ಕೊಂಡ್ಮೇಲೆ ಅನುಮಾನ ಪಡಬೇಕು ಫಾಜ್ ಅಟ್ಯಾಕ್ ಆಗಿದೆಯಾ ಅಂತ. Straight away we should not come to a conclusion ಅಥವಾ should not begin to worry, ಫಾಜ್ ಅಟ್ಯಾಕ್ ಆಗಿದೆ ಅಂತ. ಫಾಜ್ ಅಟ್ಯಾಕ್ should come at the last ಅಂತೀವಿ ನಾವು. ಇವು ಯಾವೂ ಆಗಿಲ್ಲ, ಇವ್ಯಾವೂ ಜವಾಬ್ದಾರಿಯಿಂದ ಯಾವುದೋ ಕಾರಣಗಳಿಂದ ಆಗಿಲ್ಲ ಅಂತ ಬಂದಾಗ ಆಗ ಫಾಜ್ ನಾವು ಗಮನ ಇಟ್ಕೋಬೇಕು. ಮತ್ತೆ ಎತ್ಕೋತೇನೆ ಫಾಜ್ ಅಂತ ಅನುಮಾನ ಪಟ್ಟ ಕಷ್ಟ ಆಗುತ್ತೆ ನಮ್ಗೆ.
ಸರ್ ಇನ್ನೊಂದು ಸರ್, ಕಾಲೇಜ್ ಮತ್ತೆ KM ಜೊತೆ ಸ್ವಲ್ಪ ಬಾಂಧವ್ಯ ಬೆಸಿಬೇಕಾಗಿದೆ ಸರ್.
ಇದಾರೆ ಬಿಡಿ, ಈಗ ನಮ್ ಗ್ರಾಜುಯೇಟ್ಸ್ ಕಾಲೇಜಿಂದ ಎಲ್ಲ ಬಂದಿರೋರು ಕೆಲಸ ಮಾಡ್ತಿದ್ದಾರೆ.
ಮುನ್ರೆಡ್ಡಿ KMF QC ಇದ್ದಾರೆ, ಸೊ ಯು ಕ್ಯಾನ್ ಟೇಕ್ ಇನಿಷಿಯೇಟಿವ್ ಸರ್.
ಡೆಫಿನೆಟ್ಲಿ.
ಹಾ ಸರ್, ಹಂಗಾಗಿ ಏನಾಗುತ್ತೆ ಸರ್, ಕಾಲೇಜ್ ಮತ್ತೆ KMF ಜೊತೆ ಒಳ್ಳೆ ಸಂಬಂಧ ಇದ್ರೆ ಸರ್ ಭಾರಿ ಅನುಕೂಲ ಆಗುತ್ತೆ ಸರ್.
Sir, if the KMF comes forward, we are ready to take, sir.
ಈ ಸಾರಿ ಇನ್ನೊಬ್ರು ಬರೋವರಿಗೆ ಕಾಯ್ತಾ ಹೋಗ್ಬಿಡಿ, ನಿಮ್ ಕಡೆಯಿಂದ ಸ್ವಲ್ಪ ಪ್ರಯತ್ನ ಹಾಕಿ, ಯಾಕಂದ್ರೆ ಮುನ್ರೆಡ್ಡಿನೇ ಇದಾರಲ್ಲಿ.
Yes, sir. Yes, sir. Yes, please.
Thank you ವೆರಿ ಮಚ್.
Thank you, Rudraina.
Others also, if you want, you can interact. Tirumales is there, I think. Shanta madam from Mother Dairy is there. Shubha, if you want to interact with sir, you can interact. We have 5 minutes time.
ಹೇಳಮ್ಮ
Ready, sir?
ಅನ್ಮ್ಯೂಟ್ ಮಾಡ್ಬೇಕು ಶುಭ.
Shubha, you have to unmute. Mic. You can ask, you can interact with.
ಹೇಳಮ್ಮ, any question?
Shubha, you can talk to sir.
ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಸರ್.
ಚೆನ್ನಾಗಿದೆಯಮ್ಮ?
ಹಾ ಸರ್, ಚೆನ್ನಾಗಿದ್ದೀನಿ. ನೀವು ಸರ್?
Sir Munreddy, sir. Rajaratnam sir want to talk, sir. Yes, sir, Rajaratnam sir.
Okay, Dr. Shankar sir,
Yeah.
I'm going to ask some generalized question apart from bacteriophage. See, the industry is now having a big problem because of this, coming for the plant source of milk and other things. People are getting distracted, particularly youngsters.
Mute madla.
So, can you give some idea how we can able to tackle this one, sir? It's a general question. Or you can tell me later or through WhatsApp also. Because
That's a good session.
Sure.
And because there are people who would like to know more about phages and things like that.
Sure.
So your question is a little away from the main topic.
Yeah,
So
yes.
but if you send me a query through
Sure.
WhatsApp,
Thank
I'm more than happy to share whatever little knowledge I have on that.
Thank you.
Okay,
Thank you.
Dr. Ratnam.
Thank you.
Thank you, Dr. Ratnam.
Thank you, sir.
If there is no question to Dr. Shankar sir, then we will move to the next technical session.
Dr. Ramchandra, good morning.
Yes, sir. Yes, sir. Sir, you can talk to Shankar sir, sir.
Sir, good morning, sir. Manjunath here, sir.
ಚೆನ್ನಾಗಿದ್ದೀರಾ?
ಚೆನ್ನಾಗಿದ್ದೀವಿ sir. How are you, sir?
Oh, fine. Thank you, Manjunath.
Sir, to add up one more point for application of bio, this phages, and off late around 1.5 year before, I had a sample which came from some Belgium. It is used for the removal of the biofilm in the sterilizers.
Okay.
And I couldn't get the complete depth of the microbiology, what is involved in that. But they were very clear that, sir, this product is promoted as we have used the phages to remove the biofilms which has been caused. This can be used in dairy industry also, even in the fruit juice industry also. And this is one of the promising antimicrobial activity. And this can be used in clinical, veterinary and food safety, even in environment, killing of the environment pathogenic bacteria also. And this is one of the application which has been recently, which is coming fast. Still, I couldn't able to go ahead with it. But still, as a trial basis, we consumed it, and the results were really good when we went for the microbiological testing after the CIP. It was really very good, sir. Thank you, sir.
Okay, thank you.
Manjunath, you can speak to sir. Honestly, we have interact with sir because a lot of places are there, sir. In the webinar, he'll give one or two chances. Manjunath. Okay, if there is no question to Shankar sir, then we'll move to the next technical session. I would like to thank Dr. B.S. Shankar sir for giving us the presentation and information about the bacteriophage. Thank you, sir, for your nice presentation.
Thank you.
Open it.
Of bacteriophage of lactic acid bacteria. Thank you, Chaitra. ಆಗಲ್ಲ ಇದ್ದಾರೆ ಆಯ್ತು ಬರ್ತೀನಿ. Hello, good morning to one and all.
So almost my topic has been taken care by my mentor and guide, Dr. Shankar. One thing I just want to tell about this is many must be having that question in their mind, especially KMF people. So it's starter failure is because of only bacteriophage. It may be sometimes wrong. So it may be because of inhibitory agents, like as he told about this antibiotic residues, maybe during mastitis and all, there may be a lot of antibiotic residues coming. That may be one of the factor. Second one, too much of subculturing of the same culture may lead to loss of this plasmid. Plasmid is one thing which has partially controls the lactose metabolism. So like transportation of the lactose, then hydrolysis of lactose, so then galactose catabolism, all these things may be responsible. And once the plasmid is lost, there may be chances of that bacteria losing the production of this lactic acid. So at that time, I think you have to go for a new culture. So that may help you. Okay, I just took a deviation. Actually, my topic is nothing but the morphotypes of the bacteriophages of lactic acid bacteria. So Bradley has given, next. Yeah, many things already you know. All of them told it's bacteria eater and all these things. I don't want to stress the same thing again. So now we have the two phases. One is the bad side and another one is the beneficial. So this bacteriophage, next. So there are 10 to the power of 31 bacteriophages identified. And some of the important events include this Hankin, who started this. He saw this Ganga water, so was free from bacteria, and he got to know that there may be some agent which kills the bacteria. So at that time, it was not known as bacteriophage, and the agent was passing through the filter. That was one thing. And second one was from that is achievement by Twart. So he also saw this small agent that infected and killed bacteria. And he had the doubt whether it is from the bacterial enzyme or from any other agent that destroyed this bacteria. So the last one I've taken is Harrell. So he also called it as invisible because it was unable to see under the ordinary microscope. So it required the electron microscope. So but it was giving the antagonistic effect on this bacteria. So that's why finally they were known as bacteriophages, which infect the bacteria. Next. So this bacteriophage structure and all already has given. So just to summarize, it has a head and a tail, a contractile sheath, base plate, and even the spikes and all. So in some of the morphotypes, it may be missing. So when I go to tell that, so I'll give it in detail. So next slide. Yeah, this is the taxonomy of bacteriophage of lactic acid bacteria. So in this, so they first started with International Committee for Taxonomy of Viruses. So they kept this realm in the sense highest taxonomy of this, that they call it as called by the ICTV. So in this, the families are important. The 4 families for lactic acid bacteria include Myoviridae, Cypoviridae, Podoviridae, and Innoviridae. So some of the characteristics I'm going to tell in the next slide. So in the first one, you can see it's a regular bacteriophage structure, what we have understood and what we keep it in our mind. So that is icosahedral. It's not all the bacteriophages of lactic acid bacteria are non-enveloped. There is no envelope for that. So envelope means there will be another layer which may be made up of lipoprotein and all. So that is missing in this. And apart from that, it has icosahedral head having 20 facets or the faces and a long tail. And also it has 6 types of these spikes or what we call fibers, which are kinked. And I think I've given about this KB and all, that is kilo base pairs. 33 to 240. And it can make around 40 proteins. And GC ratio also they have given for this myoviridae. So that is approximately around 35%. So under this examples comes as RZH, C10. These are the codes actually. And this will infect the host, Lactococcus lactis, subspecies lactis. So coming to the second one, Sipoviridae, it belongs to Bradley's classification B. Actually, Bradley has given 7 classification. Under that, our lactic acid bacterial phages fall into only 4, A, B, C, and F. So this also is non-enveloped. As I told, only difference here is in the prolate head. That is nothing but longish, the extended phase of the head. So, and it has this, the size, width, length, and all it has been given, 55 to 65, 40 to 48, and doesn't contain the non-contractile sheath. Actually, contractile sheath helps in injecting the DNA into the host. But this lacks that, but even then, so they're able to send the DNA inside. And one thing about bacteriophage is, so once it injects the DNA, its job is over, the DNA will take care further. And the ghost remains outside the cell during the lytic cycle, like that. So, or even in the lysogeny. So it has a long tail. So I found that this, whatever the prolate head, the longish head phages, they produce more of lysine, it seems. That is somewhat the comparison of the morphotype with their activity. So all of them have double-stranded DNA. So except the last one, I think, it's 50 kb, that is kilo base pairs it has, like 80 GC and all. So in the GC content is approximately 50%. So coming to the next, for this, so this P50 and this CH2, so which infect the Lactococcus lactis, subspecies lactis, bulgaricus, and Streptococcus thermophilus. And one more thing about this is they require a specific receptor. Unless that receptor is there, so they cannot adhere to the host. So that we call it as specificity of the bacteriophage. Next, coming to the Podoviridae. So you can see very small head and the tail, non-enveloped, icosahedral. So around 60 nanometer diameter of the head, contractile sheath it has, short tail, very short tail it has, and it has that is double-stranded linear DNA, and G plus C ratio is around 65.2 to 65.4. So it also has 6 fibrils or the spikes. So that will help in adhesion, and these spikes normally look into a particular receptors. It may be calcium, it may be ribitol that is present on the peptidoglycan. So that help in the actual adhesion. So there are 2 actually, irreversible and reversible. I think next speaker, so he is renowned speaker in the microbiology, especially dairy microbiology, S.K. Tomar sir. I think he will give you information in detail. So regarding this, the code is KSY1, so that infects Lactococcus lactis, subspecies cremoris. That's what I'm telling. This C10, which infects lactis, won't infect the cremoris. That specificity is there with the bacteriophage. And the last one is Inoviridae. So this Inoviridae is also non-enveloped. As I told, all of them are non-enveloped. Filamentous, you can see tubular structures, and it is length here 700 to 2000 into 7, that is diameter. It has a fibrous tail. This is having single-stranded DNA, standard DNA. It's circular. Others have the linear, that is Podoviridae is linear, others circular, and GC is 40.5 to 60.7%. That is the GC ratio. And in this, only one is that is code is B6, which infects Propionibacterium fridendrici, that is subspecies Shermani, so which is used in the preparation of Swiss cheese or Emmental cheese preparation. Next. So the same thing I have depicted again. So how this myoviridae looks, Sipoviridae. One is actual, that is line figure, line diagram, and followed by the micrograph, that is electron micrograph. Sipoviridae, Podoviridae, and Innoviridae, you can see the filamentous one. Yeah, to conclude my topic, so these are very important with respect to lactic cultures. So because lactic cultures we use for fermented milk products like curd, dahi, etc. So they are specific, I told you, regarding receptors and different morphotypes. What I discussed is A, B, C, and F of the Bradley's classification. And they may be having icosahedral and folate hydrid. And depending on the morphotype, you can relate. So once you see the activity, you can see like producing more of lysine and all. So tail is lengthier or it may be smaller. Sometimes contractile sheath present or absent. So the morphology of the fudges, it has already understood. It may be related to the activities. Thank you very much. Next.
Thank you, ma'am. May I now kindly request Dr. Malashri madam to introduce our next speaker, Dr. S.K. Tomar sir, to the audience.
Yes. Good morning, sir.
Hi. Good morning, Malashri.
How are you,
Can
sir?
hear me?
Yes, sir. Yes, sir.
That's fine. Good.
Okay. So I am profusely elated to take an opportunity to introduce Dr. Sudhir Kumar Thomas, sir. So he was a former principal scientist at NDRI in the division of dairy microbiology. So sir has worked as scientist in the department, and then he has worked as in charge of National Collection of Dairy Cultures and Techno-Functional Status Lab, Electron Microscope Center, and also he has worked as academic coordinator at NDRI, ICAR NDRI Karnal. So his specialized area of research includes bioprospective novel and native strains of lactic acid bacteria for their conservation as a national bioheritage and characterization to produce functional biomolecules like vitamins, mannitol, trialose, GOS, tagatose, bioactive peptides, bioaccumulation of selenium, zinc for probiotic attributes, and to study their stress biology, and also has worked in the development of technologies for functional dairy foods. And he has carried 30 plus research projects, both at national level and international level, and some of the national funded projects at ICAR, DBT, DST, MOFPI, ICMR, and also Nestle India. And some international funded projects are from BBSRC UK, India partnering, and DST, Mobi, New Zealand, Work Bank, Samsung South Korea. Approximately has guided 38 research scholars, and he has published more than 300 papers in peer-reviewed national and international scientific journals. And sir has received many awards, and to name few, he has received Indian Council Agriculture Research Best Teacher Award in the year 2018, and he has also received award from Commonwealth New Zealand Postdoctoral Fellowship in the year 1984 to 86, and also received award from United Nations Development Program Fellowship in the year 1981 to 83. So once again, I welcome you, sir. And so we are very eagerly waiting for your presentation, sir. And please, you can carry with your presentation, sir.
Please,
Hello, sir.
hand over me the presentations. Make me host.
Okay, sir.
Okay, sir.
You have to make me host.
You will only change, sir?
You will.
No, yeah, I will handle it. Yeah.
Okay, sir. Thank you. Thank you.
Thank you. Wait a minute. Can you see the slide, please?
Yes, sir. Yes, sir.
Okay. Very good afternoon, Dr. Shankar. Good morning from India. Dr. Prabha, Dr. Ramachandran, and Dr. Malashiri. And there should be some Shilpa Sri also somewhere around. Yes, I have two alasses in my life. I could never meet Dr. Shankar and another Dr. Prabha. So anyway, today I'm fortunate to see them at least, to share the dais in webinar. And Dr. Shankar made my job quite easier, and rather he explained better my content. So my presentation will be simply a supplementary material or extension of what Dr. Shankar told. And rather, I would say my presentation will be more a Bollywood film. Bollywood film has a lot of masala, don't make any sense. So may it be the same here. Anyway, when Dr. Prabha told me to deliver a lecture, so I thought of viruses, how we can describe them. So two words came to my mind, Machiavellian and thrifty. So let's see what they are. The dictionary meaning of Machiavellian is a personality who is very cunning, manipulative, and exploit others. So I think this is what viruses do. And this is the picture of that Machiavellian original person. After him,
Sorry to interrupt you, sir. Sir, please share full screen, sir.
It's not coming complete?
No, sir. No, sir.
Oh, wait a minute then. I have to stop sharing and...
Full screen.
Yeah, I don't know why now it's coming. I see the full screen anyway. Let me see. Yeah. Okay. Oh.
Full screen ಮಾಡಕ್ಕೆ ಇರುತ್ತೆ. ಇರ್ಲಿ ಅದೇ ಹಾಕಿ.
ಚೆನ್ನಾಗಿದೆ ಅದು ಪೀಪಾಲ್ ಬಂದಿದೆ 22% ಬಂದಿದೆ 2% ಬಂದಿದೆ ನೋಡ್ದಾ
Now it's coming?
Yes, sir.
Now.
Okay, fine.
Yes,
Yeah.
yes, sir. Coming.
Okay.
Yes, sir.
Actually, all these different online platforms, they have different mechanism. So anyway, yes, I'm aware of this. So yeah,
Yes, sir. Coming, sir.
mechanism is cunning. Second, what is being thrifty means? Well, thrifty means using one resource carefully, good luck, or piggy bank. Yes, so viruses, they are manipulative and they use their resources very carefully, and at the same time, they are strong and they remain healthy no matter its host becomes sick. Yes. Before I go further, let's talk about the size of the viruses. Of course, they can only be seen with the electron microscope. So I say, like even Dr. Shankar told, that they are smaller than bacteria, but still they overpower bacteria. So usually smaller the weak. No, the small can be mighty too. In Hindi, there is one poet, the old poet Bihari. He said this sloka or this thing couplet, Dekhan mein chote lage, ghav kare gambhir. In translation, they appear minute in appearances, but they inflict serious wounds. Yes, here there are three smallest viruses, and there have been like confusion. Earlier, it was on the bottom, MS2 bacteriophage, which is of size of 27 nanometer. It was considered a smaller one, but thereafter another came, this parvovirus. It was considered the smallest one. It has a 18 nanometer diameter, and now the award goes to porcine circovirus in being smallest. It is 17 nanometer. So it's just more a textbook information for the students. The smallest virus is porcine circovirus, which is 17 nanometer in its size. Then big brothers. Yes, there are viruses which can be seen with the microscope, not only electron microscope. Yes, the biggest virus is Pandora virus. Then there are three more bigger virus. Earlier, in fact, it was the other one which were bigger. Now, the recent one is the Pandora virus. So there are Mimi virus, Pithovirus, and Megavirus. Out of this, Mimi virus is known for mimicking microbes. They are so much close to bacteria, and if you gram stain them, you can do them, and they will come gram positive. They are able to retain crystal violet iodine complex inside them. Okay, coming to the T phages. T phages, don't worry to go through the detail. So T phages are of two even numbered and odd number. Even number can be T2, T4, T6, and so these are the three even numbered, and there are four odd number, 1, 3, 5, 7. Means from 1 to 7, so there are 7 phages, 1, 2, 3, 4, 5, 7, and almost alternately 1, 3, 5, 7 are the even and odd, and 2, 4, 6 is the even number. Out of this, T4 is the most studied bacteriophage. Yes, phages are studied for studying in molecular biology, microRNA, the understanding genetic code, phage therapy, nanomedicines, and drug discovery and their delivery. So these are the applications they are being used. Yes. So this is how viruses look like. This is a filamentous, like Dr. Parba told. So in appearance, it is a filamentous. They are electron micrograph. This is again complex viruses with the head and tail fibers. This is icosahedral. This is only having head and tail, and this is icosahedral. So which is the MS2 phage, and this is head and tail, which is the lambda phage. And then this is one recent paper, 2021. There is a 10 micrograph of Klebsiella pneumoniae. So here you can say again, this is a T4 phages kind of appearances. They have even the Klebsiella pneumoniae. Yeah, so phages are found in a variety of morphologies: filamentous, lipid-containing envelope, and phages with the lipids in the particle cell, as Dr. Prabha told. Their genome, yes, this is very, peculiar about phages. Phages are they are the travel light if you must. They are the best traveler. They carry the most lightweight. Usually we are supposed to contain, a biological cell is supposed to contain both DNA and RNA, but they carry only one, either RNA or DNA. They don't carry both. So they have simply nucleic acid and proteins, the minimum thing. Smaller than this are prions, which are only proteins, and there are virions, which are only nucleic acids. Otherwise, so they are the smallest thing because they only contain DNA or RNA and a little bit proteins. There are about 5,500 bacterial viruses which have been characterized by electron microscopy, and as Prabha ma'am told, this most studied group of phages is the tail phages. They belong to order Cordoviriales, and there are three types: Syphoviridae, Podoviridae, and Maiviridae. Here they are. Out of this, if you take, say, phosatic Podoviridae, it has a non-contractile small tail, non-contractile. Then Syphoviridae, they too have a unsheathed head and unseat tail, and it is again non-contractile. Myoviridae, it is the T4 phages which belong. It has a head, it has a tail, and whiskers, and seats one. Please, you may excuse me going a little bit fast because I have, as I said, it's a 3 hours Bollywood movie, so a lot many things to show you. Yes. Now coming to the T1 phages, say T2, T4, T6. Yes, these are the different stages which Dr. Shankar told: adsorption, penetration, synthesis, replication, maturation, and release. I will talk about these hurriedly. Yes. This phases looks like they watch TV too much, because they have their life cycle just of 30 minutes, like the duration of a serial we have on a TV. So they complete their whole cycle in 30 minutes, just like a TV serial. So don't go into the detail here. It is said the lytic, they have the lytic cycle, and this lytic cycle, it passes through 5 stages. Adsorption, once they enter the cell, as Dr. Shankar told, they penetrate, and after penetrating, they take over the genetic machinery of a host, and then they force their host to cook their food of their taste, that is enzyme synthesis. And then first proteins are synthesized, then DNA is synthesized, and then they assemble, and then they leave their host by killing their host. So here it is 100 to 150, but no, this number goes to 300 virion particles. There is video. Let's see. I don't know whether sound will come or not.
Bacteriophages were first co-discovered in the early...
Can you see? Are you able to hear the sound of video? Could you hear?
Bacteriophages were first co-discovered in the early 1900s by a British scientist. Scientists noticed that clear circles called plaques were forming on their petri dishes that were growing bacteria. The bacteria was being killed. Durrell named them bacteriophages, literally meaning
Yes,
bacteria.
sir. You can see video, sir. Yes, sir.
They solely rely on random encounters of bacteria that they infect. Once they come in contact with their specific host, they attach to the surface of the cell with tail proteins and begin to degrade the cell wall. They then inject the DNA with the bacteria in the head or the capsid through their tail and into the bacteria cell. Depending on the virus, the cell does one of two things at this point. If the phage is a lytic virus, the cell's machinery, including the ribosomes, will begin to make viral proteins that were coded in the newly integrated viral DNA within minutes. The cell continues to replicate the phage progeny until eventually the cell bursts open and hundreds of newly created bacteriophages are released into the surroundings, looking to infect more bacteria. If the phage is lysogenic, on the other hand, it does not burst the cell open immediately. Instead, the genetic information is replicated along with the rest of the cell's genome and remains dormant. Once the conditions are right, that's when the viral replication initiates and lysis of the cell occurs.
Okay, so this is a brief things. So first stage is the adsorptions. For adsorptions, you need the host. So there are the couple of things present on bacteria which act as a receptor site. It can be flagella, it can be pili, it can be carbohydrate or protein present on the membrane in case of animal cells, or cell wall in case of bacteria. There are specific receptor in the bacteria, is lipopolysaccharide or lipoprotein or both. For example, in case of phages T3, T4, T7, it is the lipopolysaccharide of tail fibers act as a receptor in the phages T3, where lipoprotein acts as a receptor in T2 and T6. There should be a receptor on both sides, on the viruses tails as well as on the host also. In T5 phages, both LPS and lipoprotein, they act as a receptor. When the receptor are absent, the bacteria become resistant to phage. Yes, then it is a holiday for bacteria. If the receptors are missing, it is immune to viral attack. There is another video.
The T4 phage initiates infection of an E. coli bacterium by recognizing cell surface receptors of the host with its long tail fibers. A recognition signal is sent through the long tail fibers to the base plate. This unravels the short tail fibers that bind irreversibly to the E. coli cell surface. The base plate changes conformation and the tail sheath contracts, causing GP5 at the end of the tail tube to puncture the outer membrane of the cell. The lysosome domain of GP5 is activated and degrades the periplasmic peptidoglycan layer. The remaining part of the membrane is degraded, and DNA from the head of the phage can travel through the tail tube and enter the E. coli. After the life cycle is complete, the host cell bursts open and ejects the newly built viruses into the environment, destroying the host cell.
Okay. Now, yeah. So, well, this shows how the phages, they arrive. They contract this sheath, and they hold the host with this whiskers. And when they contract their sheath, it's just like a holy pichkari. It ejects its DNA into the host with the help of kind of an enzyme like lysozyme-like enzyme, which drill through the peptidoglycan layer. And then it shows how there are different receptors present on the bacterial cell wall. And so yeah, it's yeah. So these are the receptor of different phages. For Syphovirus, it is a capsule pellicle, as well as they can adhere to flagella. Podovirus and myovirus, they adhere to more valticoic acid on bacterial cells. Again, this is from a research paper. So yeah, as you know, these are the three kind of viruses, and these are the host receptor. In case of gram-positive, there is a narrow range of a receptor like this, while in case of only one kind, while in case of gram-negative bacteria, it can be two, one kind of receptor or two or more receptors. They are present here. Yes, once bacteria, they land on the surface, they do the pinning. The second stage is known as the pinning. In case of pinning, they squeeze this tail and they inject the DNA into this. It's like this. It's like this series, and the sheath contracts from say 24 discs to 12 discs to inject its DNA. Yeah. Then once it is injected the DNA, the phage head and tail, they remain outside. Only the DNA goes inside. I mean, phages are very, professional. They know, what to be done. So, yes. So this is how they look like. Before they penetrated, they had the DNA here. After DNA, now they are in the empty head. Sometimes it looks like more like a weight loss exercise, like we have pictures now. Before you take our therapy, you look like this. After our therapy, you are like this. So yes, after injecting the DNA, now the virus is relaxed. Now it is thinner than before. Yes, after this penetration, now it takes over. Yeah, it's like holding the host at a gunpoint. Yes. After holding taking over the molecular machinery and metabolic machinery of the host, what bacteria does? So first, it degrades, it demoralizes the host cells. So first thing is, and you remember it is a 30-minute cycle. So first the degradation of bacterial chromosomes. After infection by T1 phage, the bacterial chromosome is unfolded due to the relax of superhelical twisting. It is unfolded. The nucleotide which is present in the center is disrupted and DNA helix is attached with plasma membrane at different points. This DNA of host gets attached to the plasma membrane. This process facilitated by nucleotide disrupting gene, T4 DNA gene products. So these are the genes which forces the host DNA to attach to the plasma membrane. Then there is a RNA polymerase 1. It binds to the membrane simultaneously along with this. So this take about 3 minutes. After 3 minutes of infection, protein synthesis by bacterial host stops. No, bacteria will not make a protein for itself, but it is taken over and partially breakdown bacterial chromosome occurs. So host chromosome is broken. Now, however, up to 10 minutes, non-conservative DNA synthesis occurs just to repair the damage of the bacterial DNA caused by nuclease encoded by phage. Yes, first is, it overpowered decades. Again, then it resynthesized because it wants a DNA to synthesize the viral DNA. Yes. So, first the DNA degrades the host. Second is the protein synthesis. So phage DNA contains early. Now there are two kinds of proteins which are made, and for that there are two kinds of genes, early and late genes. Therefore, after a few minutes of reduction of genome, bacterial messenger RNA and protein synthesis stop for host. Early and late genes transcribe at different intervals. The early messenger RNA, late messenger RNA respectively. So these genes, they transcribe two kinds of RNA, early messenger RNA and late messenger RNA. After 5 minutes of infection, the early messenger RNA of phage has been detected. Means after 5 minutes, there is early messenger RNA. And after 10 minutes, there is a late messenger RNA. And after 20 minutes, both of these RNAs are found. Yes, again, DNA got two strands. Now the left strand of T4 DNA is responsible for early genes, and while the right strand is responsible for the late genes. And this early genes are of two types: immediately early and delayed early genes. We will talk about this. Immediate early genes are transcribed after 1.25 to 2.5 minutes of infection by using the bacterial RNA polymerase. These genes also code enzymes that degrade bacterial chromosome. The delayed early genes transcribe the messenger RNA from 2.5 to 3.75 minutes. It continues up to 4 minutes. The delayed early gene synthesize the phage enzymes which produce 5-hydroxymethyl cytosine. So this is the first phage enzyme which is produced 5-HMC by using cytosine of the bacterial DNA. At this stage, bacterial restriction enzyme could not degrade the phage genome. Before this, it was possible that bacterial restriction enzyme could kill the host genome. Now it is completely taken over. Now the late phage genes products are the lysozyme and the structural components of new phages, particles like head and tails and fibers. I will show you. They are made later on. Yes. Now this is the I would say gist of the whole presentations. So once the phage DNA reaches the cytoplasm, imagine the phage DNA reaches the part of it is immediately transcribed by the host cell RNA polymerase to synthesize, as I said, new enzymes. There are about 20 proteins which stop host cell macromolecular synthesis. They will say no host bacterial protein will be made. Then a nuclease, this 20 protein, what they will be containing? A nuclease which will degrade the host cell DNA, nucleotide which are released, they act as a raw material for making viral DNA. It will contain, these 20 proteins will contain enzyme, will break down the precursor for the normal host cell nucleotide deoxycysteine triphosphate and new transfer RNA, and that read T4 messenger RNA more efficiently. Enzyme for formation of, I told you, 5-hydroxymethyl cytosine, and in host it is the cytosine. This 5-hydroxymethyl cytosine is nothing but viral DNA, viral protein. Phage T4 DNA initially transcribed by bacterial RNA polymerase, and among the first proteins synthesized are one for the modification of host RNA polymerase, thus altering the recognition sequence so that RNA polymerase no longer bind at the normal sequence of host DNA. So that RNA polymerase, which was supposed to bind to the bacterial DNA, it doesn't bind there. Rather, it binds to the phage DNA for phage replication. So now many viruses do not carry genetic information even for enzyme which are to be transcribed. In that case, they ask the host DNA to provide these enzymes too. Then there are the late proteins. I said like early proteins and late proteins. The early proteins are 20. Then late proteins are about 87. The part of the DNA which was only part of the host DNA was transcribed for early protein synthesis, which was not transcribed earlier. Now that is transcribed, and that make proteins. They code for the proteins. There are 55 proteins for head and 32 are for tails. So now these proteins are synthesized. Yes. So in replication of this, now next like protein synthesis, next is the DNA. So there are these 5 stages: degradation of the host DNA, synthesis of 5 HMC I told, prevention of incorporation cytosine into the T4 DNA, glycosylation of T4 DNA, and enzymology of DNA replications. Yes, during the first 10 minutes after infection of the phage, DNA, no phage could be recovered from the infected bacterium. For first 10 minutes, no phage is made. This time interval is called as eclipse period. Actually, as we know, this phase cycle, growth cycle is known as the one-step cycles. So first there is an infection period, which is accompanied by the eclipse period. There is no increase in the number of viruses. During the eclipse phase, no infectious phage particles can be found either inside or outside the bacterial cells. Phage nucleic acid take over the host biosynthetic machinery. Early messenger RNA code for the early and late, I told. After synthesis of phage DNA, late messenger RNA, late proteins are synthesized. This I told. And then, yes, we can focus on this table. At 0 minute, adsorption takes place. At 1 minute, first inhibition of synthesis of host DNA. I told DNA and RNA and protein all. Then after 2 minutes, synthesis of first messenger RNA. After 3 minutes, degradation of bacterial DNA. 5 minutes, initiation of synthesis of viral DNA, synthesis of messenger RNA. Then completion of synthesis of head and tail. I told up to 10 minutes, no viruses can be detected. But yeah, by the 12 minutes, it is over. By the first commercial interval in, now this is a time for first commercial interval of TV serial. And now it's ready. The assembly is ready. Now, 15 minutes, the first phage particles appear, and 22 minutes about bacteria lies, and about at some places 150, at some another places 300 progeny phages, they liberate out the host cells. So this is all said here. There is one video here.
In a bacterium infected by T4, new bacteriophages are assembled in a stepwise process. The shaft builds up, DNA is replicated, and the prohead assembles as an empty shell. But how does the DNA get into the prohead? The initiation of DNA import is not entirely clear yet, but once DNA, the packaging motor, and prohead interact, the DNA is rapidly threaded through a pore in the circular motor at the speed of about 2,000 base pairs a second. Once the head is full, the packaging motor cuts the DNA and the complex folds of an expanded head. The shaft and long tail fibers are attached to complete the infectious particle. Within 1 hour, more than 100 new phages are released from a single infected bacterium, which makes the T4 phage one of the most efficient but also fascinating killing machines.
Yes, then last is the assembly, and I told like in some after 12 minutes it's start making, and 300 new phages are made, and Yeah, I will show the assembly video also. Yes, the last is the lysis. There is a lysozyme-like enzyme is made, which break down the peptidoglycan from inside and cell lysis, and they get out. And this is the one step which I was telling. The first is the inoculum, and the second is the eclipse period. Third is the burst size. That's why it is known as the one step cycle. Yes, here is a video where there are different stages of synthesis of this thing are told of. I wish it could go back. So first stage is the base plate synthesis, and then second is the tail fibers and sheath synthesis, and then the head is made and DNA is inserted. So I continue this video. No sound here. This is tail sheath is being made. There are certain tail proteins. Then head is made. There is a receptor on the head. Now, there is assembly, the tail, which has been separately made, base plate, then the tail, it is going to attach to itself. Already DNA is already inside. No, sorry, this is the DNA being inserted. Excess DNA is removed. It's a perfect tail ring. And now other proteins comes, and now the tail will be attached. Some whisker on the collar, whisker protein around the neck. This is the envelope being made along the capsule. Different envelope proteins. So it contains DNA. So now tail comes from, which has been separately synthesized. It comes. So whiskers are being added, and DNA is ready along with this bow and tie to go to the party and to infect the another host. Okay. Yes. So as Dr. Shankar told, there are two phages, lytic phages, lytic cycles and lysogenic cycle. In case of lysogenic cycle, the viral DNA does become a part of this thing, the host DNA, and it doesn't lyse. But at some point of time, there is a breakup, and they break their path, and they again become the lytic cycles. So, few terms for the students. When the bacteriophage enter those phases which enter into this lysogenic cycle, they are known as the temperate phases. Prophage is that viral genome which is present in the bacterial chromosomes, and lysogen is the complete together is known as the together the bacterium temperate phase, that becomes the lysogen. So this is how it can enter, and this is again small animations. So prophage is a part of the host DNA, and together it is known as the lysogen. At some point of time, as Dr. Shankar told, that the UVA radiation, etc., it breaks away and it starts behaving again like lytic cycles. So this is what I told right now. So actually there are this lambda phage is example of this. So they have a this lambda phage is a gene C1 which produces the repressor protein and a receptor site to which receptor can bind. When this site unoccupied, DNA replicated, protein formed. When occupied, no replication and gene expression takes place. So there is a race between this receptor site and this C1 gene. Yes. Now, when I was preparing for this, I found a third way. So like, okay, lytic cycle, lysogenic cycles. There is another cycle, multiple cycles. You see this picture from movie Sole. Here you see Dharmendra and Amitabh both traveling together in the same mobiles, either motorcycle or this attached this thing. Yes. In lytic and lysogenic cycle, only one virus multiplying, but there are chances where two viruses can infect same host and they can together multiply. They are such a great companionship. So they are great friends as well. So this is a third type. Usually in textbook we talk of lytic cycle and lysogenic cycle. No, there is a multiplicity cycles as well. This is in the next slide. So you don't read this because this PPT will be given to you. I mean, organizer will be sharing with the participants. So here it is being showed that like when you, and it happens like when UV dose are given. So when there is a alone viruses, it is a low survival. Alone viruses, that is monocomplex. When there are two viruses which host, which infect the cell, then they have better survival. So they help each other. So again, this is in case of UV dose, and this is in case of what we call MMC dose, that is mitomycin. So when mitomycin is given, so in that case, when there are two phages inside, they opt for this multi-complex survival, and you see it's far better as compared to the ekla chalore or the monocomplex alone this thing. So this is the third cycle. Yes, I know I'm running out of my time, so I come towards my end. Yes, there is one Robin Sharma famous book. Dr. Shankara surely must be aware. The monk who sold his Ferrari. So this virus is a great combination of a monk as well as a criminal. He is a monk who carries the gun with him. So this monk, he carries very, few things with him, but he doesn't sell his Ferrari. He rob the Ferrari of his host. So this is what I'm trying to say. And well, thanks to everybody. Thanks to the organizers who gave this opportunity to be on a same platform where the great luminous scientists like Dr. Shankara is there. And I need to it ends. Bye.
Thank you, sir. Thank you for your elaborated presentation, sir. Hello?
Yeah.
Any questions from the audience?
Yeah, I miss you. I remember when you your whole batch came to our house.
So, any questions from the audience?
I don't think they understood anything. It was so hurried up.
So, any questions from the audience? Okay, if no questions, so once again I thank you, sir, I just recalled your class at NDRI after a long time, sir.
Yeah.
And it was nice.
Yeah. I used to teach viruses in team 611 to you.
Yes, sir. Yes, sir. Exactly. So it was nice listening to your lecture, sir. Thank you so much, sir.
Thank you, sir.
Thanks, Dr. Prabhat. Thanks, Dr. Ramachandran.
Thank you, sir.
Wherever you are.
Yeah.
Thank you, sir.
Nice to, yeah, someday I will come to your college. I would like to come to your college.
Sure, sir.
Sure, sir. We'll be waiting.
Can I see Shilpa Sri if she is around?
Yeah, she is in chemistry, sir. We are
No,
microbiology.
but her account is being used by Malasri. I think Shilpa Sri, some name is here. Is the same Shilpa
Yes,
Sri?
she has joined, sir, for the webinar. She has joined as an audience.
Okay, fine.
She's an audience for the webinar, sir.
Okay. Right, so I got
Thank you,
this.
sir. Yeah.
Okay. Then.
It's great meeting you,
then.
sir. Thank you.
Okay then. Have a nice day. Bye.
Thank you, sir.
Thanks again.
Thank you, sir. Thank you, ma'am. I now call upon our senior PG student, Bhargavi, to introduce our next speaker, Dr. Vishwanath Angdi, sir, to everybody.
Good morning to everyone, respected dignities and all the students. Myself, Bhargavi, senior PG student. I am here to welcome Dr. Vishwanath Angadi, sir. Presently, he is working as Assistant Professor of Food Microbiology in College of Agriculture, Hassan, University of Agriculture Sciences, Bengaluru. He holds doctoral degree in Dairy Microbiology with MSc Dairy Science in Dairy Microbiology. He possesses 10 years industrial experience at various capacities, majorly in ITC Foods in Bangalore for 7 years. He is handling National Mission on Food Processing project funded by Ministry of Food Processing Industries as principal investigator and also handled 2 university-funded research projects as co-principal investigator. I now call upon Dr. Vishwanath Angadi, sir, to address the session. Hello, sir. Sir, please unmute. Sir. Sir, please unmute, sir. No.
Angdi, unmute. Unmute. Voice not coming. Voice. Angdi, voice is not coming. Voice. Unmute. ಕಳ್ಸಿದ್ಯಾ ಮೆಸೇಜ್ ಮಾಡ್ಬೇಕಷ್ಟೇ ಅದರಲ್ಲೇ ಇಲ್ಲಿ ಮೆಸೇಜ್ ಕಳಿಸ್ಬೋದಲ್ವಾ Unmute. Speaker. ಕಾಣಿಸ್ತದೆ.
Okay.
Okay, ಇಲ್ಲಿ ಇದ್ ಮಾಡ್ಬೇಕು.
Sorry, there is an interruption with the microphone.
It's okay. ಇಲ್ಲೇನೋ ಮಾಡ್ಬೋದು.
Good morning to everybody. Myself, actually, thanks for the introduction, Ms. Bhargavi. My topic is based on the sources of your the. Just a minute, I'll share the slides. Being able to see the contents?
Yes, sir. It's coming, sir.
Yeah, thank you. So I thank the previous speakers, Dr. Shankar, my mentor, and Dr. Tomar for giving me opportunity to.
Make it full screen, sir. It's not visible.
Sorry?
It's not visible, sir. Full screen. You have shared, sir, but it is a.
Sir, can you please start the slideshow?
Yeah, given the slideshow only.
Sir, sir. You have shared another this one tab.
Just a minute. Enter screen. ಅದು ಓಪನ್ ಇದೆಯಲ್ಲ ಸರ್ ಇದೆ ಒಂದು ನಿಮಿಷ Is it being shared now?
Make it full screen, sir.
Now?
No, sir. Can I operate from here, sir?
Yeah, it's better otherwise. Because share. ಇಲ್ಲ ಅಲ್ಲ ಅವ್ರು ಮಾಡ್ತಾರೆ Yeah, thank you. Next time. So let me start again. Sorry for the again interruption with the technical glitches. So my topic is basically on the bacteriophages as a hidden enemies. So what are the sources of these bacteriophages in the dairy industry, and what are the consequences that will happen during their attack in the dairy fermentations. Next slide, please. Next slide. Yeah, it is just a brief note. Already the earlier speakers have already given you enough about bacteriophages. Then what are the resources, that is sources of this bacteriophage in the dairy environment, and what are the symptoms of the phase affected fermentations? Like if the phase get attacked in the fermentations, how do the product look like? Then the consequences or the impact of these phase contamination in the dairy fermentations. Next slide, please. So here, as the earlier speakers told, the phages are the viruses that infect bacteria. So they have been already told like phages are the bacteria eaters. So there on the Earth, a study shows that there are about 10 to the power of 32, 10 to the power of 32 total phase particles on the Earth and the biosphere. Okay, so they outnumber the bacteria by 10 folds, as Dr. Shankar was telling. So wherever the bacteria with a sensitive bacteria for the particular phase, then there will be a particular phase. So almost 100 numbers means if 1 million is there, they may become 100 million. So that is the one. So the predominant, they are basically the predominant entities, biological entities in the earth. Like almost you can say, if you take a single drop of seawater, it contains about 10 to the power of 8 phases. Okay, so they are almost present wherever the bacteria are found. So in case of dairy fermentations, the first the bacteria, that is a Bacteriophage, was noticed in 1935 by Whitehead and Cox. So later on, several Bacteriophages were found, and these lactic acid bacteria phases belongs to the family Caudoviridae. So in that we have seen other orders like different type of Bacteriophages. So basically, they are double-stranded DNA packed in the head-on with tail. Okay, and the phase infection is the most common cause for the slow as well as incomplete fermentations in the dairy industries. Okay, so this implication of this phase attack, so basically it happens to the financial losses. So because of the product loss or because of the raw material loss, we'll be losing the amount here. Okay, the next slide. So when you come to the source of phase contamination in the dairy environment, so basically there are we can classify the contamination into two types of contamination. One is primary contamination. So that means it lays outside the facility. Means it may be a cheese factory. So it not exactly, it comes from the external source. That's why we call it as outside the facility, and they are present in low quantity. Okay, the number of phases will be low in quantity. So they are like raw milk and ingredients, whatever the ingredients we use. In case of milk fermentations, we use mainly the raw milk. In the raw ingredients, it can be the other things which are added. It may be sugar or it may be skim milk powder or other things. Then personal, the people who are involved in the production of these products, they are considered as personal. In the case of secondary contamination, so they are basically previously contaminated sources. Means it can be a processing line which are already having the bacteriophages, and they'll be present in a large quantity compared to the primary contamination. Okay, so here it is mainly we get is the factory environment, air or surfaces, basically the surface of the equipment, and the phase reservoir. So phase reservoir is nothing but the reservoir of the phases, basically in the inside the facility. Okay, the most important thing is the recycling of the weigh products. So that is also one of the source of these phase contamination. Next slide, please. So coming to the first source, that is the raw milk. The raw milk, like we collect the milk from different societies. Okay, so from the different suppliers. But the biodiversity of the phases get amplified in your silos or the cheese vats, because whenever we get the phase sensitive bacteria, they will get attacked, and the phase numbers will get multiplied. So as such, the raw milk contains about 10 to the power of 4 plaque forming units, PFU. We mentioned for the bacteria or the microorganisms, we give the unit as CFU, but here for the phase, we use the term plaque forming unit. So which you can see here. So this is the plaque forming units where there is a clear zone around the colonies. Okay, and the phase titers of about 10 to the power of 6 to 10 to the power of 7 PFU per ml is a significant value which can lead to the fermentation failures. Okay, so some studies have shown in the Spain, like from the different geographical area of the Spain, they've collected the milk samples and they checked for the phases. They found that 10% of the raw milk samples were affected with the Lactococcus lactis phase. Okay, and in the same Spain, they have found that the milk samples which are used for the production of yogurt, so there are about 37% of the milk samples were affected with the lactococcal and streptococcal phases. Okay, so but we may say that we are going to pasteurize that, how it will affect that, but pasteurization temperature is largely insufficient because at that temperature they may not get affected. So to eliminate these most of these lactic acid bacteriophages, I have 2 slides in the coming thing, so which it will mention about their thermal resistance. Okay, so some studies say that from the idea of that longer time treatment, that is about 90 degrees centigrade for 2 minutes, is required to kill the phages. But the same study also say that there are some few exceptions where 90 degrees centigrade for 15 minutes is also required to kill these phages. Next slide, please. Then coming to the personnel or the workforce. So it's the people involved in the handling of the raw milk. So basically they carry the phages basically on the aprons, on the hands, on the whatever the personal protective equipment they possess. So based on that, they can carry the phages. And they are people involved in the cheese making process. They are the indirect contacts. The workforce involved in the wire handling, that is in the recycling. So they also carry the bacteriophages. Then contamination during the subculturing. So if you're not following the good laboratory practices, so if you're not sterilizing the loops or the petri plates or test tube properly, that may lead to the contamination. Then people who are handling the products, the cultures, like the poor personal hygiene or improper hygiene of the vats used in the cheese making. So they also lead to the because of these negligence. People neglect negligence. They don't wash the equipments properly. They don't adhere to these phase control steps or strategies. Because of that, also it will lead to the contamination. Next. Then the factory environment. So here it is the environment in the particular factory, that is the cheese making or the starter culture area. So where during the CIP or during the addition of the milk or transferring the milk, there will be a liquid flashes or the air displacement around the vats for the cheese preparation, or some contaminated surfaces in the vats or in the knives which are used. So when the splashing occurs or the air displacement occurs, they generate the aerosols, that is the tiny droplets which also carry these viruses. Okay, and these viruses, they will remain in air for a longer period. Okay, and they become the major cause or the root of phase dissemination from one place to the other place. So that is the aerosolization, like this distribution of the aerosols, that is dispersion of this is referred as aerosolization. So that is the important route of dispersion in the cheese industry as well as the fermentation industries. So air contamination. So basically the phase counts vary from 10 to the power of 2 to 10 to the power of 8 PFU per meter cube, especially in the cheese plants. Okay, so you can imagine they can affect the sensitive lactic acid bacteria. Then we have various devices for analyzing this air contamination, but they damage the virus structure so that it will give the false negative results. So this is one of the drawback in analyzing the contamination of air with the phase particles. So they develop in the places like in the equipment surface where we are not able to access those places. Like there may be some pipelines or some places where we can't access for cleaning and disinfection. So in that case, they will multiply there if they found the favorable host, that means the sensitive bacteria. Okay, so it has shown that in one of the study that air sampling for 12 hours in a cheese production line, they have found about by qPCR, that is a quantitative PCR test. So they have found lactococcal phases, that is 936 and C2-like phases, like about 10 to the power of 3 copies of DNA per meter cube, they are present. So the adequate ventilation is required for the prevention of these phages. Next slide, please. Then the recycling of the whey and whey products. So we know that we use the whey. We don't, most of the times we drain it to the this thing, but here we use the whey as a whey protein concentrate, mainly to improve the texture of the food, to increase the nutritive value, to increase the yield of the product, and for standardization of the products. So here it shows that the cheese whey contains about the 10 to the power of 9 PFU per ml of phase counts. Okay, so these phases which are present in this byproducts, that is a whey or a whey protein concentrate, so they contaminate the products. Okay, and these phases remain in the whey even after heat treatment of 95 degrees centigrade for several minutes. This is because there is an encapsulation effect, that is a fat and protein and lactose. They protect the phages from the thermal damage. So this is also the external protection for these phases. Okay, and in case of natural fermentation, the whey contains the phases of the serious threat, because they never add. So we should they will have the very strange phases. So that's why we should never add the whey products from the natural fermentation, because from the known cultures we know the exact the microflora, and we can also control those phases. But the unknown cultures, so they may be the severe threat or a serious threat for the dairy industry. And the WPC usually often contain the high temperature resistant phases. So they survive and cause the contamination. So some of the phases which are highly resistant to the heat, so they also cause the contamination. And you also know that that is the process of microfiltration and ultrafiltration is used in case of the whey treatment. So there the whey get concentrated. So that will lead to the increase in the whey count, that is a phase count, about 1 log. So they also remain in the places of membranes of MF and UF, so that becomes the contamination source. Next slide, please. So there is a term called as phase reservoir. So phase reservoir is nothing but the known or earlier reservoir of the phases. So we call it as lysogenic starter cultures. So the earlier speakers were telling me that the prophase. Prophase is nothing but the phase DNA incorporate into the bacterial DNA. So that is intact, and it gets transferred from one generation to the other generation during the multiplication. So the lysogeny here means there is a prolonged harmony with the bacteria. That is, the phase DNA remains with the bacterial DNA for a longer period. So that's what we call it as a lysogenic cycle. Okay. So the factors like heat, salts, antimicrobials, starvation of the bacteria, or UV rays, they can trigger this lytic cycle, and that will lead to the natural induction. So that is so this is, it can reach up to 9%. In 9% of the cases can happen when get exposed to the factors like this. And the recent study shows, spoken to you by the next speakers. So these are the majorly this includes the 3 things. One is the source of phase contamination. So as I already told, raw ingredients, the equipment surfaces, staph, air, and bacterial strains. Then other things like the phase detection, like what are the different methods of phase detection, and what are the phase control strategies. So which will be known to you in the next presentations. Okay. So I think this is the information which I wanted to give. Thanks for providing me the opportunity to share my knowledge in this webinar. Thank you.
thanks from all of us, sir. Now we have our next speaker, Dr. Ramchandra, sir, to speak on various techniques involved in the detection of bacteriophage.
Sorry, mute.
ಯಾವ್ದೋ ಬೇರೆದಾಗ್ತಿದ್ಯಾ? Okay. Again, good morning to all. I will be presenting the detection methods of forge detection methods. Different detection methods of forge are there, but I will present only the golden standard method to detect the forge by using DLA or double layer agar method. Next question. The plate method, it is also called as double layer agar method. In this, the number of flakes are determined by the double layer agar method. That is the golden standard method for detecting the forge, and it is also one of the traditional method to detect the infected forges in the any fermented milk products or waste samples. One more method is there, that is acidification method, where the difference in acidity or pH is taken as an indication of the infection of forge. As far as the other methods are concerned, let's go to next slide. There are other methods like the transmission electron microscopy, TEM method is there. Then flow cytometry method is there. Then nucleic acid based methods are there. But I will not go in detail of these advanced methods. I will concentrate on the DLA double layer agar method, forge flake method, and the acidification method. Next. You can detect the forge infection in a number of samples, particularly in case of fermented milk products, like the whey sample. You can detect the forge milk, then cream, curd, yogurt, and sour cream. Then starter cultures can be the starter cultures which are contaminated or infected with the forges can be determined. Then unknown cultures also can be determined for the infection of forge. So before detecting the forge by the double layer agar method, you have to prepare the samples from the product. So in case of whey samples, if you take the whey samples directly for the detection of forge in the whey samples, so the whey samples will be collected from the different tanks or from the incubation of the fermented milk products, then we can be tested for the infection of forge using the double layer agar. So the preparation of whey sample, the whey samples will be diluted 10 times or 1:10 dilution will be done using peptone water. Then filtration of the diluted whey sample to remove the solid residual particles to obtain the clear whey. Then the whey obtained is collected in the sterile test tube or in the vial. Then it can be refrigerated or stored for until the forge infection, until the detection of forge in the given whey sample. So in case of yogurt, fermented milk products, sour cream, and bulk starter cultures samples, again the same method of sample preparation, like dilution to 10 times. Then using peptone water, again dilution will be done. Then has to be subjected for centrifugation. Centrifugation can be done at the RPM of 6000 for 15 minutes. That will give the separation of supernatant and the sedimented residues. Then the supernatant is collected, then this can be treated as a waste sample for the infection of FOS. In case of curd samples can be also tested for the FOS infection, but you have to do the pre-treatment like homogenization, mixing of sample. Then again, the sample of whey can be obtained from the curd or dahi, like in case of the yogurt or any fermented milk products. You have to dilute the sample, then centrifugation, then collect the sample and treat this waste sample or collected supernated as a waste sample for the infection of FOS. Then how to prepare the culture for the to detect the FOS infection in case of the starter culture. Starter culture may be the frozen starter cultures or any liquid starter cultures can be detected for the FOS infection. So take the culture from the deep freezer, then melt it, then subject it for melting or thawing. Then mix the culture with the 10 ml of reconstituted skim milk. Then incubate for curdling. Then in case of mesophiles, that incubation temperature is 25 to 30 degrees centigrade, whereas in case of thermophiles, 37 degrees the incubation to obtain the firm set curd. For the enrichment of FOS, if the FOS number is very low, then you have to do the enrichment of the FOS. The enrichment steps in case of the FOS enrichment, take the sample in the sterile test tube, then rinse with the swab in case of the swab used for the sampling, then rinse the swab in the peptone water. Then add 1 ml of filtered sample. Then to this add 10 ml of reconstituted skim milk. Then to this add 0.1% of overnight milk culture. Then incubate the sample for setting. Whereas in case of if there is no proper acidification of the culture, we can do the acidification of the culture. If there is no acidification, you can go for the acidification of the culture. Then subject the sample for the centrifugation to collect the supernatant and treat this supernatant as waste sample. Next. So in case of the double layer agar method or flake method, the steps in case of double layer agar method, the preparation, dilution of overnight culture, as I told in the earlier slides, you have to prepare the culture dilution. Then you have to prepare the agar. Then if number of cultures are given for the testing, then you have to name the number of strains in separate test tubes. And the order of preparing the sample for the double layer agar FOS detectance test, take 0.05 ml of 1 molar calcium chloride. Calcium chloride facilitate the absorption of the FOSs to the bacteria, helps in absorption. Then take the culture to be detected for the FOS, 0.2 ml of first diluted or 1:10 diluted mesophilic or thermophilic culture. Then to this add 3.5 ml of molten overlay agar. The overlay agar may be M17 in case of lactococci or Streptococcus thermophilus, or MRS in case of Leuconostoc or Lactobacilli. Then mix the content using the vertex mixer. Then transfer this mixed sample onto the already solidified agar plate. So first you pour the agar to this sterile Petri plate, then allow the media to solidify, then pour this overlay agar containing culture as well as calcium chloride. onto the solidified medium. Then allow this overlay agar to get solidified. Then this will become a double layer. Then once the plate is ready, after proper drying, you can dry the plates after pouring the overlay agar by incubating in the refrigerator. Then take the sample, waste sample, to be detected for phage infection. So if the number of samples are more, then you can make the boxes, different squares on the agar plate, or only one sample is there, means you need not to go for the making of square boxes on the agar. If the number of samples to be tested are more, you can use the same agar plate for number of samples to detect the phage infection. Okay, next. So this is the spotting of the sample to be detected for phage infection. So take the waste sample, then spot on the overlay agar. You can take 5 to 10 microliter of the filtered waste sample, or the waste sample which is prepared from the any curd or yogurt or any fermented milk product, or from the acidified cultured or starter cultures. You can take the waste sample. So this waste sample is used for spotting on the solidified overlay agar containing calcium chloride, as well as the culture to be detected for phage infection. Then incubate the plates overnight. In case of mesophilic, 30 degree. In case of thermophilic, 41 degree centigrade is the incubation temperature. Then after the incubation, then observe for the formation of flakes on the agar plate. Next one. So this is the, so in case of number of samples is more, then you can make the square boxes on the same agar plate. You can go for number of sample more than one, means you can go for the number of samples on the same plate. Make the squares on the agar plate. Then you can spot the waste sample to be tested for the phage on each boxes. Then the right side shows the infection of phage. So the cleared zone indicates the infection of phage with the particular starter cultures, or any waste sample is taken, means that particular sample is infected with the phage. That will give the clear zone or clear formation of agar that indicates the infection of phage. So each phage will give the one flake formation, then can be counted as number of flakes formed on the media plate. So here also, if the dilution is done, then accordingly dilution we can mention on each clear zone or flake colony formation. You can count the number of flakes like colony forming unit. You can count the flake forming zones on the agar. So in case of the acidification test, another method of detecting the phage infection. So again, preparation of overnight cultures. Remove the culture from the deep freezer, then thaw or melt the culture, incubate the culture, then preparation of test sample, centrifuge the culture, collect the waste sample, then you can do the serial dilution of the sample up to liquid. If the number of doubtful phage infection is more, then you can go for the higher dilutions. Then milk to be prepared for the acidification. 60 gram of skim milk powder is dissolved in 500 ml water. Then overnight culture, prepared overnight culture is added to the sterilized reconstituted milk, then kept for incubation. So after the incubation, the milk will be tested for the acidity or pH level. Next slide. So you can go for the number of dilutions. So take the number of sterile conical flasks, then keep the control without added whey sample, then according to the number of dilutions made, then add the culture and keep it for acidification overnight. Then after the fermentation, you can determine the acidity level or pH level in the fermented product. If there is a difference of more than 0.2 pH, then it suggests that or indicates that there is a phage infection in the sample. That is the acidification method. The difference in acidity or pH of the control as well as the whey sample added test sample. If there is a difference in pH of more than 0.2 unit or the difference in acidity indicates the infection of phage, because the phage, in case of infected phage, the acidity is reduced. That's why the reduced acidity or difference of acidity between the control and tested test sample or whey doubtful whey sample added to the milk will give the difference, and this difference can be taken as a indication of phage infection in the given sample. That is the acidification method. So these two test methods are normally used in the laboratory to detect the phage infection in the known culture or unknown culture. But particularly known culture, it is very easy to detect the phage infection in case of known culture, because these phages are specific for the cultures. Then if you know the cultures, then it is easy to detect the phage infection in case of particular or specific cultures. The other methods also available. As I told, the transmission electron microscopy can directly detect the phage presence, and also flow cytometry can be used to detect the phage. Then the nucleic acids. Right away now we are using for COVID virus detection the PCR method, that also can be used to detect based on the molecular test protocol or nucleic acid protocol. Okay, thank you.
Thank you, sir. So far, we have understood what is the bacteriophage and how they infect the bacteria. The most important aspect to know is how do we prevent them. Here comes our next speaker, Dr. Malashri Madam, to give us some hints on how do we control the bacteriophage in fermented dairy industry.
Good afternoon, everyone. So fermented dairy products have been an important component of nutritional diet. So when this father of fermented milks, Eli Metnikov, has suggested to consume the fermented milk with the lactic acid bacteria, so then the quality and safety of starter. So with this background, the topic I'm going to present here is the control of bacteriophage in fermented dairy industry. We know that the definition of straticulture and all, so like these are the selected strains of lactic acid bacteria that are used to ferment the milk with the desirable characteristics like acid production, flavor production, and as a result, this will contribute to the body and texture characteristics also. And hence, the straticultures are said to be heart of the fermented dairy industries. And there is a enemy for this straticulture, that is bacteriophage, which attacks the starter bacteria. And these bacteriophages are the primary cause for fermentation failure in the milk transforming dairy industries. And these bacteriophages are very small in size and spreads easily, and they are also heat resistant. And even they can survive the routine pasteurization temperature of milk, and to some extent, when they survive this heat treatment, it becomes very difficult to eliminate the bacteriophage. And cheese is one of the most important fermented dairy product and is said to be a living food which contains the starter bacteria either of bacteria, starter types either of bacteria or even the fungi. And the cheese industry with the whey separation, okay, so the whey will be a major source for this bacteriophage attack. And if we don't control this bacteriophage attack in the starter culture room or in the dairy industries, so then it becomes a incident and it leads to economic losses. Hence, we need to understand, or we need to study the different methods used to control the bacteriophage in the dairy industries. So some of the general methods to control the bacteriophage in lactic acid bacteria includes usage of phage inhibitory media, where the media will be made deficient with the calcium component, as this calcium component helps in attachment of the bacteriophage to the cell wall of the lactic acid bacteria. And then we have this early renneting method. So during the cheese preparation, after addition of the starter cultures, so within the 30 minutes after the addition of the starter culture, it's better to go for renneting. So because after the rennet is an enzyme which consisting of rennin and pepsin, so this helps in coagulation of the milk, leading to the formation of coagulum with the calcium paracasinate, and hence there will be complete usage of this calcium component from the milk, and as a result even the milk or the whey will be made deficient of calcium. As a result, even in this way also we can control the bacteriophage. And the heat treatment, so heating the milk, raw milk at 75 degrees Celsius for 30 minutes or 85 degrees Celsius for 20 seconds will result in destruction of the bacteriophage. And the next method is rotation of the starters. So we know that the starter cultures, the bacteriophages are strain specific, and we know that there are different types of starter cultures like single strain, multiple strain, and mixed strain starters. So once we go for rotation of these starter cultures, so that will help in this shifting of bacteriophage from one culture to another culture. As a result, so there won't be any accumulation of the bacteriophage in the culture, and that will lead to avoidance of this bacteriophage. Attenuation of starters. So this is like we'll be leaving the bacteriophage to dominate or to combat with the starter cultures, and as like survival of the fittest, the starter cultures which becomes or which resist against the phage will be collected separately, and then after collection they will be propagated as attenuated starter cultures. So through this way also we can avoid the bacteriophage attack. And finally, we have this maintenance of starter room, so which is very important for all the dairy industries. So we have to take care of the complete design of the starter room and also processing line, and also there need to be adopting of this HEPA filters in the starter rooms. As a result, there is a control of aerosols which carries this bacteriophage, and the quality control manager who is working in the starter room should be free of any viral diseases, etc. And so these are some of the okay, so then we have some genetic methods to control the phages in lactic acid bacteria. So like inhibition of phage adsorption, blocking of phage DNA injection, and restriction modification system, abortive infection, and CRISPR-Cas system, antisense RNA method, and bacteriophage strike back method. So in the first method, inhibition of phage adsorption. So this is like the bacteriophage is very specific to the receptor sites on the cell wall of the lactic acid bacteria. So the one way of inhibiting the phage adsorption is through blocking the phage receptor, and the second way is by production of the extracellular matrix by lactic acid bacteria. And also we have one more method, that is plasmid encoded lactic acid adsorption inhibition in lactic acid bacteria. So in the first type, that is blocking the phage receptor, so we know that the bacterium evolves phage resistance by the modification of this cell surface receptors. So here the receptor site is modified. As a result, it blocks the adsorption of the bacteriophage on the cell surface of the lactic acid bacteria. And in this figure, you can see that even there will be mutation of the receptor site built by this lactic acid bacteria. As a result, again, there will be blocking of phage receptor to the lactic acid bacteria. And then even the bacteria is capable of producing the proteins called masking proteins, the phage which recognizes the site receptors, thus making the receptor inaccessible for the phage adsorption. And in the second method, the one more is that some bacteria produces the extracellular matrix, that is like exopolysaccharide. So once this exopolysaccharide covers the receptor sites on the cell surface, so then there is a chance, there is no chance of attachment or adsorption of the bacteriophage to the lactic acid bacteria. Even though some bacteriophages are capable of producing some enzymes which can degrade this exopolysaccharide, the enzymes called hydrolases and lyases will degrade this extracellular matrix and they'll attach to the surface. And these are some of the examples for plasmid encoded adsorption inhibition in lactic acid bacteria. So the PSK112 in Lactococcus lactis subspecies cremoris helps in ಅಲ್ಲಿ ಕಾಣಿಸಿಲ್ವಾ ಸರ್? So helps in so this the plasmid PSK 112 in Lactococcus lactis subspecies cremoris helps in masking galactosyl containing lipoic acid on cell surface and prevents bacteriophage to adhere to the receptor site, thus developing resistance. And the another plasmid PC1528 of this Lactococcus lactis subspecies cremoris synthesis cell surface antigen, that is EPS, that shields the receptors against phages. The next method is prevention of phage DNA injection. So certain lactococci develops the resistance against phage tail lysins, and so in this here we can understand that so this prevention of phage DNA can be, if phage DNA injection can be achieved through this superinfection exclusion system. So here first the attachment of phage receptor on the surface of the host cell takes place, and there will be an inner membrane protein that aids the translocation of phage DNA into the cell cytoplasm. And later on this inner membrane protein directly blocks the translocation of the phage DNA into the cytoplasm of the phage. Later on, there will be involvement of surfactant protein, that is SP protein. So this phage DNA is prevented from entering the cell cytoplasm by inner membrane protein and also surfactant protein that prevents the degradation of the peptidoglycan layer by inhibiting the activity of the lysozyme. The third method, that is restriction modification system. So in general, so in case of lactic acid bacteria, so there will be, this system can be achieved with the help of majorly two enzymes, that is restriction endonuclease and also methyl transferase. So here the restriction endonuclease recognizes and cuts the unmethylated double-stranded DNA at specific recognition sites, whereas methyl transferase adds methyl groups to the same bacterial genome recognition sites on the bacterial DNA, and then it protects the self genome from restriction endonuclease cleavage. So in this figure, we can understand the restriction modification system by another method, another way, like so the phage's passive and active strategies to bypass restriction modification system. In the first figure, we can understand that this RM system is achieved by the phages that possesses few restriction sites in their genome, are less prone to DNA cleavage by the host restriction endonucleases. And in the second figure, we can understand that so occasionally the phage DNA might modified by bacterial methyl transferase upon successful injection into the host, and again this will also helps in avoiding the bacteriophage attack. So in the third figure, we can understand that some of the phages, while injecting the DNA into the host cell, it also co-injects host genome binding protein, that is DAR A and DAR B, that mask the RM recognition sites. And some phages possesses protein that can mimic the DNA backbone, and a protein that mimics the DNA phosphate backbone and has a high affinity for the RM system. So these are some of the examples for restriction modification system characterized from Lactococcus lactis and Streptococcus thermophilus. And the one more method, of genetic engineering is that abortive infection. So it is abbreviated as ABI mechanism that includes broad range of defense that can interfere with genome replication, transcription, translation, packaging or assembly of the whole phage particle. So and then become an important play and come into play after the injection of the phage DNA. So once this complete reactions starting from the replication to the packaging of the assembly is restricted, so then there won't be any multiplication of the infected bacteriophage, and around 23 ABI systems have been identified till date in lactococci, ranging from ABI A to ABI Z, and most are plasmid encoded. A single gene is generally responsible for the phenotype, and although the involvement of two genes have been proposed. So this is again, this diagram helps in understanding the abortive infection in case of lactic acid bacteria, and this is achieved by two components of Rex system. So that is Rex A and Rex B. So the phage protein DNA complex activates the sensor protein Rex A, which in turn activates Rex B. And then Rex B is an ion channel that causes depolarization of the bacterial membrane, leading to the cell death. So some more new methods are already in field, like the antisense method, phage therapy, and also phage immobilization, etc. So, but till date, majorly these four methods, like inhibition of receptor site, RM system, abortive infection system, are majorly used. And to conclude the presentation, so as I've told that this sanitation, process chain, strain rotation, and use of this phage resistant strains, these are all common control measures to be taken at the level of dairy field. And then this lactic acid bacteria are still today a serious industrial concern, and manufacturers are constantly waging war against these viruses to keep them under control. So in spite of these efforts, phages are evolving and new variants keep emerging. So thus, it is necessary to find novel control and antiviral strategies to keep up the phage evolution. Thank you.
Thank you, ma'am. That was really helpful. We have been seeing bacteriophage as an enemy. It's time to see bacteriophage as a potential friend. Here we have a next speaker, Dr. Pradeep Behre sir, to throw some light on the aspect. Before that, I call upon our senior PhD student, Rajshekhar, to introduce our next speaker, Dr. Pradeep Behre sir, to all.
Good afternoon, all. Good afternoon to all. This is a great opportunity to brief about Dr. Pradeep Vishnu Behere sir, who is working as a scientist, a senior scale, and in charge in a national collection of dairy cultures, NCDC, in Dairy Microbiology Division, ICAR, National Dairy Research Institute, Karnal, Haryana, India, from 2011. He is a graduated in dairy technology from the College of Dairy Technology, Pusat, Maharashtra, in 2001, and completed his master's from SMC College of Dairy Science, Anand, Gujarat, in 2004. He did his doctoral work in dairy microbiology from National Dairy Research Institute, Karnal, Haryana, in 2008. He serves to industry like Amul Dairy, Anand, Gagjit Industries, Kapartala, Punjab, and Wadilal Ice Cream Plants, Bayasar, Maharashtra. He worked as assistant professor dairy microbiology at the College of Dairy Technology, Udgir, during 2009 to 2010. He completed research training on mannitol production for the fructophilic lactic acid bacteria and also isolation of bacilli phage from the environmental samples at Tegas Food Research Center, Moorpark, Cork, Ireland during the June to October 2019. His research areas are isolation and characterization of lactic acid and probiotic bacteria, production and characterization of functional molecules from the lactic acid bacteria such as exopolysaccharides, vitamins, and low-calorie sugars, proteomics of probiotics bacteria, isolation of phase of dairy applications. He has made a significant contribution to the bacteria isolation of phages for a dairy application. He had made a significant contribution to the field of dairy science. He has developed and commercialized cost-effective technologies in bioprocess of for the dairy direct fat set cultures, mishti dahi, sour dahi, low-fat dahi, and low-fat lassi. He is also a part of a national collection of dairy cultures, NCDC, which is a repository of the dairy starters in India. He has handled externally funded projects from DST, SERD, MF, PI, ICAR, etc. Dr. Behera sir has published more than 80 research and review articles in high impact journals. He has written a textbook on dairy microbiology for graduate and postgraduate students. He is a recipient of several oral and poster present awards, while his contributions is recognized by the India's best academic like National Academy of Agriculture Sciences, New Delhi, National Academy of Dairy Science of India, Karnal, Society of Uplift of Rural Economy, Varanasi International Life Science Institute, ILSI, India 2021 award. I welcome you, sir, for the presenting seminar. Sir, will you operate or shall I?
Yeah, I will. Thank you very much. Shall I operate? Okay. Yeah, I can do that. Yeah. I'm just sharing my slides. Is it not coming? Hello?
Yes, sir.
Am I audible?
Yes, sir. Yes, sir.
Okay. Can you see my slides?
No, sir.
Just a minute. Now? Hello? Now can you see?
Yes, sir. Yes, sir.
Yeah, okay, fine.
So, if possible, make it a full screen, sir.
Yeah, is it not full screen?
No, sir.
Yeah, at my end, it is full screen. Anyway. There is some noise from this audio. Maybe sometime it happens. Just a minute, okay. Now can you see?
Yes, sir. But
now it's okay?
Now it's okay, sir.
Okay, fine. Sorry for this disturbance. Okay. A very good afternoon to one and all. Organizers for this webinar on bacteriophages, enemy or ally. Dr. Prabha, Dr. P. A. Shankar, and I could see some of my teacher as well from Maharashtra. So I'm going to talk about the very interesting topic, phages for control of spoilage causing bacilli biofilm in dairy industry. So since morning, I listened a lot of discussion on the bacteriophages, especially their control in the starter cultures, and how the bacteriophages can affect fermentation industry. So a lot of things we have heard negative sides of the bacteriophages, but on the other side, I can just present a positive sides which now people are looking for from these bacteriophages. This will be the outline of my presentation. So I'll be focusing on mainly the dairy spoilage causing bacillus species or the geobacillus species. Then I will also give a light on which type of properties are essential in the bacteriophages so that they can be utilized as an effective agent. Some phages which has been reported for these cultures, then how they can be used to remove various spoilage causing biofilm. And we have also at NDR have been doing some work on this line. So I will give some thought on this and what kind of results we are getting in that. Finally, I will give my conclude and give some what are the major issues or challenges when we are going to deal with this issue, this area. Now we all know that these bacillus, they are the very important spoilage causing agent. Beside geobacillus also, they are the main culprit for the spoilage of milk and milk product. And most oftenly bacilli, they have been reported from raw milk or the processed milk, and a geobacillus from the heated milk or the dried product. And once these organisms are present, they can cause a variety of changes, lipolytic changes, glycolytic, and cause spoilage of the product. Since these groups are also producing spores and they are quite heat resistant, and also they can produce a biofilm in the dairy environment, because these organisms favor milk environment because they find their this is ideal medium for their growth. So these bacilli and especially the geobacilli, so they can enter into the dairy environment and they can form biofilm whenever they get an opportunity. So where as of now, there are 3 types of biofilms have been reported for such kind of bacilli and geobacilli: pellicle, biofilm bundle, and the submerged. In the pellicle and the biofilm bundle developed generally in the industrial storage and piping system, whereas surface biofilm, this is sometimes submerged biofilm, which is at the in the bottom and which is very difficult to remove by a cleaning agent. So this is the very important aspect whenever we're talking about the biofilm formation in the dairy industry. So what is the impact of the biofilm? When the biofilm form and it is remained for longer duration, and subsequently when we are going to use the product for processing, the spectral cells which are attached in the biofilm are leaching or coming out into the product. They can have adverse effect on the organoleptic properties. They can reduce the shelf life, can increase the corrosion rate and hamper heat transfer rate, and ultimately have a significant economic losses. Although there are current practices of using CIPs and some biocides, but these agents are also not sufficient to completely remove the biofilm which is formed by some bacilli. Even some report says that now even some species are going to become resistant for certain sanitizers, and that will be the cause of concern. Alternatively, there are a number of biological molecules are coming up. There are different peptides. People have tried bacteriocins and many more. Among these, bacteriophages can be looked as one of the promising solution, because bacteriophages they are host specific, and in some cases they can also act against the multi-species organism, multi-species bacteria. And one more point is that bacteriophages, they are the natural enemies of the bacteria, and they do not cause any issues with the humans or the animals and the plant. And that is the reason that much researchers are talking about the phages and the phages therapy and the phages uses to control different types of microbial species. But before understanding the bacteriophages as an biocontrol agent, So what things needs to be understand in the bacteriophages? The first and most important thing, when we talk about the starter cultures, we do not want the virulent phages. But here, when we are looking for these bacteriophages as a biocontrol agent, we need to look for a virulent phage which can show a lytic activity. Further, in the case of starter, we do not want the broad host range phages, but here we do require the broad host range so that multiple strains or the species can be targeted. And these phages should be easily propagated that we do not want in our starters. We are looking for the phage as a biocontrol agent, their whole genome sequence should be completely elucidated because it's very important, and phage genome should not contain any virulent gene, otherwise it will again will be problematic. The phages, whatever are available, should be stable over a long period of time, and they should be stable during the application as well. The phage product should be amenable to scale. Yes, this is very important. Only the phage application should not be restricted to the laboratories. It should be further enhanced at the large scale or in the commercial level. Then how these bacteriophages can act against various kind of biofilm? When the biofilm is produced, it is very rigid or tough because of the molecule called we call hexapolysaccharide or polymer or polysaccharide, which hold a different bacterial communities together, and over time this becomes very hard because it's a very complex. So bacteriophages will attack, will produce some enzymes as well, and they will cause the degradation of this hexapolysaccharide or polysaccharide through different ways by producing some enzymes, and ultimately this bacteriophage will get chance to enter into the bacterial cells, and then eventually it will cause a lysis of the cell. So in this way the bacteriophages can act on the biofilm. There are different other strategies by which the bacteriophages can be applied. If we feel that the biofilm is of multi-species, there are different species, then fast cocktails can be used as an effective biocontrol agent for multi-species associated biofilm. Even some fast-based enzymes, endolysins, they can be also used, like also depolymerase. These enzymes can be applied, which can be useful to remove the biofilm. People have also tried to use phages with even some known other antimicrobial agent just to increase the effectiveness of the treatment. By genetically modifying phages, people have increased the lytic spectrum so that these bacteriophages, what is going to be applied, should kill the bacteria in the biofilm in a more robust way. Also CRISPR-Cas, this also can be used to again improve the phage lytic behavior. So far, when we talk about the bacilli phages, there are few phages have been reported when we compare with the other phages like Escherichia coli or Listeria monocytogenes, because these are generally considered as spoilage causing organisms. So not much emphasis has been given, but now the time has come, so we have to look for when the quality is at most important. So now the time has come, we need to focus on this spoilage causing organism also, because they are causing a huge economic losses. So some very few phages have been reported for Bacillus leukiniformis, Bacillus cereus, subtilis, and if you see, most of the phages are obtained from the non-dairy sources, and usually we say that the phages from whatever the sources they are obtained, they will be more applicable to that particular sources. So in case of Bacillus, and even in the Bacillus stereothermophilus, these are the thermophilic organism, and very less number of hardly hundreds of phages have been reported, but that too in the only for the research purpose. So very less number of phages are available. Now research are coming up, and new bacteriophages are being isolated for those even thermophilic Geobacillus species also, so that in the long term they can be applied to control these very thermotolerant organisms. Now coming to some studies on the biofilm removal by the phages or phage cocktail. So some studies have been carried out with the, as I said earlier, a lot of studies have been carried out, but with the other species, other students like Listeria monocytogenes, Staphylococcus, or aureus and Escherichia coli. So people have improved the people are removed very successful in removing the biofilm form on the some adhesive materials, well maybe the stainless steel coupons or polystyrene or other materials where these phage cocktail or phages have shown very useful effect. Either they have completely eliminated the biofilm or reduced the log count in the cells present in the biofilm. Some of the studies which I have found, I have come across, I'm going to present it here, and how they have utilized the bacteriophages for elimination of this bacilli. So one preparation called as FBL1, which comprises more than 10 bacteriophages in the preparation, and when they applied FBL1 preparation against the Bacillus licheniformis, and when they add it at 0 hour, then after producing the biofilm at 24 hours and then 48 hours. When this FBL1 preparation was added at 0 hour, it was able to, it was inhibiting the biofilm formation, or on the other side, it was killing the plankton, the newly formed cells. While once the biofilm is formed, the effect of FBL1 was little bit on the lower side. As we can see, after 24 hours, there was half of the effect could be observed, and when the biofilm which was a 48 hour was treated with the FBL1, which was not even the biofilm was remained as such. There is another study which shows the biofilm formation, inhibition of biofilm formation on UF membrane. In this case, these authors have used 3 different species which can form biofilm, and also 3 different phages. So the in one they have used these 3 different species added into the effluent. Then effluent was added with these 3 bacterial species, and one group was with the 3 bacteriophages. And what they found, when these species were passed to the membrane, these phages were able to kill these cells where their permeability was very high as compared to the effluent, which was containing only the cells. So they have mentioned that permeability drop of 84% in the control where phages were not applied, and approximately 29% with the phage treated effluent. So it means these phage preparation were very effective in killing of these 3 different bacterial species. Another one study called carried out by Shin, they have isolated 2 bacteriophages, BPS10C and BPS13, from the food waste sample. And what they have interestingly found that this BPS13, both the phages were very lytic, but BPS13 was more lytic as compared to the BPS10C, because this BPS13 genome had few prominent genes which were encoding for the endolysins and the 2 tail lysins. So because of this feature, this BPS13 has shown a more lytic behavior, and this BPS13 was also evaluated for the stability against different pH and the temperature, and this was very stable at wider pH as well as at the temperature. When we talk about the bacteriophage, and then subsequently there's some certain product enzymes. So a number of endolysins have been isolated from the various bacteriophages, and their enzymatic activities have been identified. And what literature says, these are the endolysins. They are the enzymes which have been acting against different bacillus group like Bacillus cereus, Thuringiensis, and Bacillus anthracis. That means that endolysin can also be used as one of the agent to remove the bacterial or spoiler causing biofilm. Here, one study shows that in which the endolysin MEBA2446, which was basically obtained from the Bacillus anthracis, which was produced in the Bacillus cereus. In this case, the extracted MEBA2446, when purified and added at different concentration in a Bacillus cereus cells. So these endolysins were able to increase or they have decreased the log cycle by 3 to 4 for these Bacillus cereus strain. Even they have again checked the lytic activity against different Bacillus cereus, anthracis, and Bacillus thuringiensis strains. So this was showing a very broad spectrum activity even against multiple species. There is another study which has used the recombinant endolysin called as lyse BPS 13, and they have treated this. They have used this lyse BPS 13 against the Bacillus cereus ATCC, and their study shows that where this lyse BP 513 endolysin was used at 5 microgram, it was able to kill these Bacillus cereus cells. Whereas where the treatment was not given, Bacillus cereus, this strain was growing. So it means that recombinant endolysins have also been used, can also be used to effectively remove this kind of spoiler causing organisms. These bacteriophages have also been used in the processed milk, and researchers have got very good result for killing of various spoilage causing or even pathogenic species in the pasteurized milk, UST milk, or the other commercial milk. There are some phage-based products are also available, and mainly especially recommended by FDA also for their use in the food additives. Few more are also coming up in the market. So many are in the in the stage of patent granting stages. So number of phage products are being coming up in the market, and they will be very much will be in the at our end. Now coming to the work which we have carried at our NDRI, because this Bacillus and the Geobacillus species which I have mentioned earlier, they were the most important spoilage causing organism for milk and meat product. Then we thought to isolate the phages for these species and to look for their control in the dairy industry. To begin with, we first started with the Bacillus licheniformis and Geobacillus stereothermophilos, 2 species. So again, to understand, as I said earlier, if you want a bacteriophages for a particular species, so we have to explore that particular source. So here, considering that these Bacillus and Geobacillus from dairy sources, we initially begin with the isolation of these isolates, Bacillus licheniformis and Geobacillus stereothermophilos, from the dairy sources from our NDRI. And then we went on after isolation of these cultures, we went on for the isolation of the bacteriophages. Bacteriophages were isolated by plaque assay as well as the spot assay. So isolation of bacteriophages is not sometimes very easy, especially when we are looking for the specific bacteriophages. Phages are everywhere, but to getting a proper phage is very difficult. So we have to try several methods so that we can get the appropriate phage. We have what we could find 10 bacillus for Bacillus licheniformis and about 27 bacteriophages for Geobacillus species. Then our task was to look at their lysis profile, because more the phages are lysis, then more will be there will be more effective for removal for killing of the spoilage causing organism. So lysis profile was checked, and based on the zone, type of zone, we can say that the phage which can show more zone will be more effective. Accordingly, out of the 10 bacteriophages what we have isolated for Bacillus licheniformis, the first named as ADM2 form the larger plaque size, and we feel that this is one of the promising bacteriophage maybe in the coming time to control the Bacillus licheniformis. This is the slide showing the lysis profile for the Geobacillus stereothermophilus, here GBS11. So we have given some in our internal coding, and this phage has also shown a very interesting lysis profile. Then another aspect when we are looking for the phages or phage-based preparation to kill to various polyphasic organism, the broad host range. So the phages which will be having broad host range will be more effective. So here we evaluated our all the other phages, and against different hosts we used multiple hosts: Bacillus licheniformis, Megatherium, Bacillus subtilis, Stereothermophilus, total 17 or more than that. Even we have tried, and we have used, and we tried different phages against those hosts, and we could find the BL Bacillus licheniformis phages. They were showing broadest. Many of them were showing the broad host range, infecting different types of strains as well as species. Also, Geobacillus stereothermophilus was also showing the similar kind of result. So here, to summarize this result of our research, so what we found that JBS11 phage, which was initially obtained from the ETP section of the our institute, was able to infect more number of hosts. From BL, ADM4 was able to infect more number of Bacillus licheniformis, and other even Bacillus cereus were infecting different multi-species hosts. So what we are further carrying out the research, we are now in the phase of preparing a phage cocktail where we can utilize this phage preparation or phage cocktail to remove the biofilm produced by Bacillus and the Geobacillus species. Initially we will try in the laboratory scale, and then we will go later into our experimental dairy plant where we can actually look into the effect of this formulation. Yes, in conclusion, bacteriophages, they are they show natural lysis and act against the targeted species. There are few laboratory experiments which shows the effectiveness of phages to remove the biofilm, but most of the phages are from most of the studies are conducted on the non-dairy based bacilli. So we need to isolate phages from our dairy sources, so which can be explored against the dairy spoilage causing or bacilli. Bacteriophages will be the one of the better option to control spoilage causing bacilli. But when we talk about the biofilm is very complex and may comprise a mixture of species, and in that case we need to develop a very robust cocktail or fast preparation which not only which not only kill the single species or multi species, but it should go across the species as well. So we need to develop the formulation in that way. So research should also be directed to formulate a broad host range fast preparation. Yes, coming to the key issues and the challenges. So the interesting point would be how much would be the stability of this fast preparation, how they maintain their stability, even the endolysins what we are going to act in, and actual application of phages in dairy setup, how it will be applied. There should not be ecological impact on the environment, in the sense that if the phages kill the spoilage causing organism, and if they want to go out of the factory, I mean, there should be the phages should not go out, rather they should be degraded. And there is again concern evaluation of bacterial resistance to the phages. But in that case, we can have an option. We can go for the endolysins or the phage derived other molecules which can act also give a better result. Finally, I want to acknowledge the Karnataka Veterinary Animals and Fishery Science University, my director, Dr. M. Chavan, my mentor from Ireland, Holly Omikolpi. So because of her only I started to working in this area. My student Sonia Ranvir, who helped me to prepare these slides. And thank you.
Hello. Hello, Pradeep sir. Thank you very much.
Oh, okay.
I'm Prabha.
Yeah.
Thank you very much, sir.
Yeah.
I think you have put a lot of effort on this. It's very informative. Thank you very much, sir. Sir?
Thank you. Yeah.
S.K. Thomas, sir. Sir. Thank you, sir. Thank you very much. Bye.
Thank you so much, sir. Now the platform is open for discussion. If any one of you have any queries related to the topic, you can just go ahead.
Shankar sir, good evening, sir, and good afternoon from India. It is nice to see you, sir, after a long time. Sir, you are muted. You have to unmute.
Yeah.
Pradeep, I don't know
Yeah,
whether you
sir.
heard me or not. It was a nice presentation. I'm glad
yes,
to know
sir.
that you're working on very interesting phages and bacilli. Very interesting. Because in the days to come, bacillus happens to be probiotic also. So people are worried whether phages can attack probiotic bacillus also. Anyway, interesting topic.
Thank you, sir.
Sir, Shankar, sir.
Helama.
Thank you very much, sir.
It's my pleasure, Amma. Pleasure to see all of
On
you
all of
again
your
and again.
cooperation, we could able to complete this webinar successfully, sir. Thank you very much.
Yeah, you have put so much of
Ramchandra
effort.
also
It has to be
thanks.
successful.
Sir, thank you, sir.
Thank you, Ramchandra.
Thomas,
Thank
sir,
you,
thank you,
Mala.
sir.
Thank you, all others.
Also helped me
Thank-
during my training in India, sir. Thomas, sir. Pradeep,
Oh, okay. Very
doing
good. Very
training.
good.
Thank you, sir.
Pradeep,
Thank you very much, sir.
Thank you very much.
So the technical session closes here. May I please request Dr. Malashri ma'am to propose the vote of thanks.
So, Thanksgiving is a time of togetherness and gratitude. I would like to express our gratitude to all the esteemed delegates of the university, Honorable Vice Chancellor Sir, Registrar Sir, Director of Research, Director of Extension, Dean Dairy Science, and Division Head Dairy Science for the presence and contribution to make this webinar a great success. I also thank and compliment all the eminent speakers, Dr. Priya Shankar sir from Australia, Dr. Sudhir Kumar Thomas sir, Dr. Pradeep Behre sir from NDRI Karnal, and Dr. Vishwanath Angdi sir from College of Agriculture Hassan for delivering excellent presentations on bacteriophage as a friend or foe. My heartfelt thanks to Head of the Department, Dr. Prabha Madam, and organizing team, Dr. Ramchandra sir and Ms. Chaitra, also Mr. Rajshekhar, who have worked a lot to make this webinar resounding success. I owe special gratitude to postgraduate students, Ganesh, Sirisha, Ramya, Bhargavi, and supporting staff of the department, and Mrs. Chaya Devi, Lingappa, and Nagesh for volunteer supports. At last, I must thank all the participants. Approximately around 135 students, participants were there, and staff of Dairy Science College, students and alumni of Dairy Science, and even special thanks to the employees from Karnataka Milk Federation for your active participation in the webinar. And then for all your cooperation and interest in making this webinar a positive success. And also, once again, I thank Chaitra and organizing team for giving me this chance of delivering the vote of thanks. And once again, I thank everyone. Thank you so much. So the webinar has been streamed in the YouTube. So you can download and you can watch whenever you want. Thank you so much.
Special thanks to you as well, ma'am, for your wonderful presentation. I believe that the webinar was profitable and fruitful for everyone. Thank you for taking the time to join us today. We are really grateful to all the speakers for demonstrating their views on today's topic. Well, everyone, an event like this cannot happen overnight. It requires planning and a bird's eye for details. We have been fortunate enough to be backed by a team of very motivated and dedicated colleagues. I thank our HOD and the whole Dairy Microbiology family for making this webinar a successful one. Thank you. Good day, everyone.